There is an animal almost everyone has seen, at least in passing, on a science magazine cover, in a viral video, on one of those internet lists of the world's strangest creatures, that has been quietly rewriting the definition of what a mammal can be. Most people recognize the body before remembering the name. Pink, wrinkled, with a pair of front teeth too large for the face that carries them, eyes so small they seem almost decorative, and a name that lands somewhere between a description and a punchline. Naked mole rat. It has the kind of looks that get a creature pinned to ugliest animal lists and used as a cartoon punchline. There is a cartoon mole rat named Rufus that an entire generation knows by name. There are zoo enclosures where children press their faces to the glass for a few minutes and then move on, because what they see is, by any human measure, very plainly not what we usually mean by beautiful. A pink, blunt, slightly translucent rodent with teeth that, in nearly every other mammal, would be hidden behind a closed mouth. That is the animal most people carry in their heads, a curiosity, a novelty, a small joke of evolution. Almost every part of that picture is wrong. The mammal most people carry in mind, warm-blooded, soft-skinned, sensitive to pain, eventually undone by cancer or by simple old age, is not the animal that lives under the dry soil of East Africa. This creature does not regulate its own body temperature the way an ordinary mammal does. It is indifferent to certain kinds of pain that would stop most animals in their tracks. It can spend roughly the length of a feature film with no oxygen at all, in a state that should be impossible for a warm-blooded body, and emerge from it without lasting injury. It can live for nearly 40 years, which is somewhere between 5 and 10 times the span of a similar-sized rodent, and across those decades, the body does not pass through the ordinary milestones of getting older. It almost never develops the disease that, in laboratory mice of the same size, would kill nearly all of them. There is a sentence the scientists who have spent their lives with this animal end up saying in some form eventually when pressed on what kind of mammal it actually is. The word mammal softens in the mouth when it is applied here. That softening is real, and it is a serious problem in biology. It is the reason a quiet, mostly unknown rodent has become one of the most studied animals in the laboratories of the world, in Chicago, Berlin, Rochester, Tokyo, Shanghai, and Cape Town. Over 100 labs now keep them. The largest single collection lives in a labyrinth of plastic tubing in Chicago, where a woman who first met these animals on a research expedition in Kenya in 1980 has been watching them, the same lineages, for 45 years. What those decades have produced is a portrait of a creature so far from the standard model of mammalian biology that several of the ordinary words used to describe an ordinary animal simply stop working when applied to this one. This is the animal worth sitting with tonight. If a slow evening with an unlikely animal is the kind of company that appeals, the subscribe button does more than almost anything else to keep work like this possible. And one question for the comments before we begin. Has an animal ever made one of the everyday categories of biology feel a little less solid than it should be? Those are the answers worth reading. Right now, somewhere under the dry soil of northern Kenya, in a tunnel 3 meters deep where the air is warmer and heavier than the air above it, a small pink animal is moving through a corridor she has known her entire life. She is roughly the size of a chocolate bar. She is, by every measure that matters, not the kind of mammal most people carry in their heads. She has no idea you exist. The country east of Mount Kenya is dry, low, and mostly empty. The grass cover is thin most of the year, the soil hard between scattered acacia trees, and the surface temperature by mid-morning climbs past 35 degrees Celsius and keeps climbing through the afternoon. The visible animals, antelope, lizard, falcon, snake, move through that heat carefully, taking shade where they can find it, drinking when they can. For most of them, the surface is the only world available, and the heat is the daily problem to be solved. 3 meters below that surface, the problem is different. The air at that depth is warm, a steady 29 to 32 degrees Celsius year-round, regardless of what the season does to the world overhead. It is humid. It is heavy with the breath of 70 or 80 small bodies that have not been to the surface and will not go to the surface in the whole course of their lives. The carbon dioxide reaches concentrations that, in a human-occupied building, would already have triggered ventilation alarms. The oxygen sits somewhere between 2 and 9%, depending on where in the tunnel one happens to be. About a quarter of what is breathed at the surface, and on the lower end of that range, in the same neighborhood as the thin air on the upper slopes of Mount Everest. The animals are not climbing. They were born here, and the colony is, almost always, in motion. The tunnel system stretches for kilometers. A single colony can excavate 3 or 4 kilometers of corridor in total length, with chambers and dead ends and reused passages branching from the main runs, all of it underground, all of it lightless, all of it at roughly the same warm body temperature. The depth of the main run is typically about 2 meters, though in particularly hard dry seasons, the deeper sections drop closer to 2.5 meters in search of soil that still holds moisture. Inside that network are specific chambers used for specific purposes. There is a nest where most of the colony sleeps in a single tangled pile and where the queen raises her pups. There are food chambers where pieces of underground tuber are stored against the long stretches between successful foraging runs. There is a latrine, a single dedicated chamber used for waste, and a separate garbage chamber kept apart from the working corridors. The animals do not need light to know where they are. They have been moving through this geometry since they were born, and the geometry, in any case, is one they have helped to dig. At one of the working faces of the tunnel, 4 animals are arranged in a line. The one at the front is pressed against the soil, her front teeth, large, ivory pale, and exposed permanently outside her closed lips, scrape and chisel at the hard earth, gnawing it loose in small fragments. The lips behind those teeth seal shut, tight, so the dirt she breaks free does not enter her mouth. She works the loosened soil back beneath her body with her front paws. The second worker, lying immediately behind her, kicks the dirt further back. The third worker passes it back again. The fourth, at the far end of the chain, ejects the loose soil up through a vertical shaft to the surface, where the cumulative day's work makes a small volcano of pale, dry earth at the lip of an opening only a few centimeters across. That volcano is the only sign from above ground that any of this is happening. It is also the colony's single moment of real danger. The vertical shaft has to be briefly open while soil is being expelled, and at that moment a small head exposed at the opening is briefly available to anything passing nearby: a rufous-beaked snake threading through the dry grass, a raptor circling overhead and watching for any movement on the soil. For that reason, there are always 1 or 2 larger animals positioned just below the opening, fully aware of what is happening at the surface, ready to bite something that intrudes. They are sometimes called soldiers in the literature, though the boundary between soldiers and ordinary workers in this species is, on closer inspection, more fluid than it first appears, a matter of size, age, and circumstance rather than of fixed caste. Elsewhere in the network, a larger animal is moving. She is longer in the body than the others by several centimeters, and visibly different in proportions, wider through the abdomen, with a spine that is, in literal millimeter measurement, more extended than the spines of the others. She is the queen, and she is patrolling. Her route through the corridors does not have a destination so much as a function. When she passes a worker in a narrow stretch, the worker presses flat to the floor, belly down, sometimes rolling onto its back with its feet in the air, and stays motionless until she has gone past. That submission posture is the most basic and most repeated transaction in the social life of this animal. It is the routine by which the queen's reproductive monopoly is enforced every time she walks the tunnels. The nest, when she returns to it, is a single living pile. 8 or 12 or 30 animals tangled together for the warmth they cannot maintain alone. Work published in 2024, watching paired animals in small laboratory chambers, found that when 2 of these animals are placed alone together in a neutral arena, they press their faces against each other, snout to snout, several hundred times across a 10-minute interval. Whatever the social function of that contact is, it is constant. They sleep touching, they wake touching, and they move through the corridors with their flanks, brushing the body of whichever animal happens to be closest. This is what is strange about the scene when watched closely. It does not read like the burrow of a small mammal. It reads, in almost every detail, like a beehive. There is a reason researchers tend to hesitate when they describe these colonies to people who have never thought about them. A mammalian society where one female does all of the breeding, where the others, of both sexes, remain reproductively suppressed for the whole of their lives, where individuals coordinate cooperative work, raise the offspring of an animal who is not their mother, and die without descendants of their own. This is the social arrangement of ants, of termites, of certain bees and wasps. It is not the social arrangement of mammals. Or, until a paper was published in the journal Science in 1981, it was not the arrangement of any mammal known to biology. Right now, somewhere in northern Kenya, that scene is happening. The queen is patrolling. The diggers are scraping at the working face. A few centimeters under the surface, a soldier is listening for any change in the small sounds at the opening above. A pile of small warm bodies in the nesting chamber is breathing slowly in air. A person standing on the surface could not survive in for very long. None of these animals know that they are unusual. None of them know that more than 100 laboratories around the world now spend their working hours trying to understand what makes the body of this small pink animal possible at all. They are simply at home. The name has been a problem from the beginning. It is not, strictly speaking, anyone's fault. The animal was first described in the scientific literature in 1842 by the German naturalist Eduard Rüppel, working from specimens collected somewhere in the Horn of Africa, and the name he settled on, Heterocephalus glaber, the smooth-skinned animal with the different-shaped head, was, in its Latin form, a reasonably careful piece of description. In English, however, the common name attached itself to the animal in the same way most common names do, by way of resemblance to creatures the speakers already knew. It looked vaguely like a mole. It looked vaguely like a rat. Its skin was visibly without fur. Naked mole rat. Almost every word in that name turns out to be wrong on closer inspection. The skin first. Held up to the light, the body of an adult is not, in fact, hairless. Running along the flanks and the back, and concentrated heavily around the face and tail, are about 100 fine sensory whiskers, short, pale, and easy to miss at a casual glance, but unmistakable under magnification or against a backlit hand. There are also short bristles between the toes, arranged in a way that researchers have for decades described as broom-like, used to sweep small fragments of soil out of the way during the constant work of tunneling. The animal does not look hairy. It is, by any honest count, lightly furred in a way that is functionally important for its life underground. Then the mole. Moles, the actual moles of European gardens and North American lawns, belong to an entirely separate group of mammals in the order Eulipatifla, the same broad lineage that contains shrews and hedgehogs. They are insect eaters. They use enlarged forepaws to dig. They are, in evolutionary terms, a different kind of animal entirely. This pink rodent of East Africa is not a mole, has never been a mole, and shares with the European garden mole only the same general solution, life underground, arrived at by a completely independent path. Then the rat. This is where the name does the most damage, because most listeners of the word will assume what zoologists once assumed, that this animal belongs to the same broad rodent family as the brown rat in a city alley or the laboratory mouse on a research bench. That assumption is wrong by something in the neighborhood of 70 million years. The lineage that contains rats and mice, the murid rodents, split from the lineage that contains this animal so far back in geological time that the common ancestor of the two groups was alive when dinosaurs still walked. The closer relatives of the naked mole rat are not in any apartment building or any feed store. They are the porcupines of the Americas, the chinchillas of the Andes, the capybaras of the South American river systems, the guinea pigs kept in children's cages around the world. These animals are part of an old branch of the rodent tree called the Hystricomorpha, the porcupine-shaped rodents. And they are, in a real and traceable evolutionary sense, the family this small pink animal belongs to. Not to rats, to capybaras. That single fact tends to rearrange how the animal looks once it has settled in. The blunt face, the proportions of the body, the way the limbs are set, the surprisingly soft and almost pig-like snout that points upward whenever the lid of the enclosure is lifted in a laboratory. None of those features were ever especially rat-like. They were misread as rat-like because the name set the expectation. In 2015, after 2 decades of accumulating molecular work, taxonomists made the final break official. The naked mole rat was moved out of the family Bathyergidae, where it had previously sat alongside the other African mole rats, and placed in a family of its own, Heterocephalidae, a family with exactly one member. The reason for the move was a measurement in the language of molecular biology of how long ago the lineages had separated. The molecular clock placed the split at roughly 31 million years before the present, deep in the early Oligocene, at a time when most of the lineages of modern mammals were still arranging themselves into the forms recognizable today. 31 million years is a long time to walk one's own evolutionary road. For comparison, the human lineage and the chimpanzee lineage have been separate for somewhere between 5 and 7 million years. This animal has been on its own path roughly 5 times longer than that. It is worth pausing on what that solitude has produced. The body that emerged from those 31 million years is, in nearly every detail, a body shaped by the requirements of a single environment, dark, warm, oxygen-poor, and surrounded on all sides by hard, dry soil that has to be moved daily in order for the animal to eat. Almost every visible feature of the anatomy can be read against that environment. The eyes are tiny, often described as nearly vestigial. Useful vision is not a priority in a place without light. The ears are external openings without the visible pinna that most mammals carry on the side of the head. A sensible adaptation for an animal that spends its day pressing soil-filled corridors against the sides of its skull. The skin is loose and folded in a way that allows the body to be drawn forward and backward in narrow tunnels without the resistance that taut skin would generate against the soil. None of these features are decorative, they are tools. The teeth, more than anything else, are tools. The pair of front incisors that dominate the face are not simply large for the size of the head. They are large for the size of the animal. They are also, anatomically, on the outside. The lips of this species close behind the teeth, not in front of them, which means that the incisors are permanently exposed to the air, and the soft tissues of the mouth are sealed off behind a clean line of enamel. The design, and the word is being used here in its loosest descriptive sense, not as a claim about intention, solves a single problem with extreme efficiency. When the animal chisels at the soil of the tunnel face for hours at a time, no dirt enters the mouth. None. The lips form an airtight seal behind the working teeth, and the working teeth do not require the mouth to be open. What makes the teeth even stranger once watched closely is that the two lower incisors are not fused together the way they are in most rodents. They can be moved independently. The lower jaw of this animal is built from two halves joined at a flexible midline, and each half carries a separate large incisor, and each of those teeth can be moved by the animal in isolation from its partner. The animal can splay them apart, can bring them together, can grasp small objects between them with the precision of a pair of chopsticks. Food is moved with them, pups are carried with them. Soldiers, when defending the colony, bite with one at a time. Researchers who have watched this behavior in slow-motion video have noted more than once that the movement looks oddly like the action of fingers on a hand. The brain that controls those teeth has been reshaped accordingly. A study by Kenneth Catania and colleagues, published in 2002, mapped the somatosensory cortex of the naked mole rat, the part of the brain that processes touch information from the rest of the body, and found something that, at first, did not seem credible. Nearly 1/3 of the somatosensory cortex of this animal is devoted to the representation of the incisors, not the rest of the face, not the body, not the whiskers, the teeth. In a laboratory rat of similar size, the equivalent area is a small fraction of that. In a human, the comparable allotment to the teeth is almost negligible. This animal feels its world, more than anything else, through the surface of two pairs of front teeth. The musculature follows. The masseter and temporalis muscles, the great closing muscles of the lower jaw, are enormous in this animal relative to body size. Estimates of the proportion of total body mass given over to the muscles of the head and jaw cluster around 25%. A quarter of the body devoted to the work of the mouth. In a human being, the equivalent figure is approximately 1%. This is not, in other words, a rat. It is not a mole. It is not naked. It is a deeply specialized member of an old rodent lineage that diverged from the rest of the mammalian family tree before there were elephants of the modern kind, before there were horses of the modern kind, before there was anything that would have been recognizable as a primate moving through the canopy of an African forest. The body that resulted is shaped by 31 million years of life underground, and almost every visible feature of it is a tool for that life. The name is wrong. The animal is something else. 31 million years ago, this lineage stepped off the path that the rest of the rodents were on. It has been walking its own road ever since. The road has led, among other places, to a body where the teeth are on the outside of the mouth, where the lower jaw moves in two independent halves, where a quarter of the muscle of the body works the head, and where, as the chapters ahead will show, the deeper machinery of the cell has been reshaped to a degree that, even by the standards of evolutionary biology, is hard to absorb in a single sitting. In the late summer of 1980, a young zoology student from the University of Cape Town climbed into a Land Rover with her supervisor and drove north out of South Africa, across Botswana and Zambia and Tanzania, and into the dry inland country of southern Kenya. She was 23 years old. Her name was Rochelle Buffenstein, although in the field she went by Shelley, and she was traveling with Jenny Jarvis, a comparative biologist who had spent most of the previous decade working on the physiology of African rodents and who was now, with a small National Geographic grant, going to look for naked mole rats in their natural habitat. Almost nothing was known about how this animal lived in the wild. A handful of colonies had been kept in laboratories in South Africa, and the suspicion had been growing slowly and without firm evidence that what was happening inside those laboratory colonies was something biology did not yet have a word for. They found their first sign of a colony near the town of Matito Ande on the edge of Tsavo National Park. The sign was not the animal itself. It was a small mound of pale, freshly excavated soil at the surface, almost identical to the molehills familiar from European gardens, except that there were many of them, distributed across a patch of dry savanna in a pattern that suggested a connected network underneath. The pattern was correct. Beneath the mounds, in tunnels that the two researchers spent the following weeks slowly excavating, was the working colony of a single eusocial mammal. The first one anyone had ever properly documented in the field. In May of the following year, Jarvis published her findings in the journal Science. The paper is 600 words long, occupies 3 pages including the references, and reports a single observation. In a captive colony of 40 naked mole rats collected from that Kenyan field site, only one female ever bred. The other 39 animals, including more than a dozen reproductively mature females, did not. The non-breeding members of the colony were performing distinct kinds of work, sorted broadly by body size. The smaller animals were doing most of the maintenance and foraging. The larger animals were involved in defense and tunneling, and across generations the same arrangement persisted. The paper used a word that had, until that moment, been reserved entirely for insects: eusocial. The naked mole rat, Jarvis wrote, was a eusocial mammal. The word landed in the field with the force of a small reorganization. Eusociality had been defined in the entomological literature, mostly by Charles Michener in the late 1960s, as the social arrangement of certain bees, ants, wasps, and termites. Animals whose colonies contain overlapping generations, cooperative care of the young, and a reproductive division of labor in which most adult members do not breed. For nearly 100 years before Jarvis's paper, the word had referred only to those insect lineages. Mammals were assumed not to do this. The combination of overlapping generations, cooperative brood care, and a single reproductive female had been observed nowhere in the mammalian world, and the assumption had hardened over decades into a quiet certainty. Mammals were not eusocial. Then this small pink animal turned up in the soil of Kenya and was. What made the moment stranger was that the possibility had already been predicted 4 years earlier by someone who had never seen the animal. In 1976, an evolutionary biologist at the University of Michigan named Richard Alexander gave a lecture at Northern Arizona University. The lecture was a thought experiment. Alexander wanted to work out from first principles what kind of mammal might plausibly evolve into a eusocial social system, and what set of ecological circumstances would have to obtain for that to happen. He reasoned his way through the requirements out loud in front of his audience. The mammal in question, he said, would have to live entirely underground. It would have to live in soil hard enough that a colony could defend its burrow effectively against intrusion. It would have to feed on large, irregularly distributed underground food items, large enough to feed many animals from a single find, irregularly enough distributed that the cooperative effort of searching paid off. The colony would have to be large, multi-generational, and reproductively monopolized by a small number of breeders. He described this hypothetical animal in some detail. He had no specific creature in mind. When the lecture ended, a member of the audience approached him. The man was a mammalogist named Terry Vaughan. He had recently visited a laboratory in South Africa run by Jenny Jarvis, where colonies of an obscure East African rodent were being kept in glass-walled enclosures for the first time. Vaughan told Alexander that the animal he had just described, in every particular, already existed. It is rare in evolutionary biology for a hypothesis and its confirmation to arrive in this order. Alexander, his graduate student Paul Sherman, and Jarvis subsequently began a long collaboration that produced 10 years later the foundational book on the species, The Biology of the Naked Mole Rat, edited by Sherman, Jarvis, and Alexander, published by Princeton in 1991. The book runs to more than 500 pages. Almost everything biology now knows about how this animal lives in the wild traces, by one route or another, back to that volume and the field expeditions behind it. What those expeditions and the laboratory work that followed them eventually made clear is that the social system of the naked mole rat is not merely insect-like in arrangement, it is insect-like in consequence. Almost every member of the colony, by some estimates more than 99% of the animals born into it, will die without ever having reproduced. They will spend their entire lives, often more than a decade, sometimes more than two, working in support of a queen who is, very often, their mother or their sister or their aunt. They are not physiologically sterile, removed from the colony, isolated from the suppressive presence of the queen. A non-breeding female will, within weeks, undergo a kind of delayed puberty. Her hormones will shift, her ovaries will activate, her body will begin to change shape. She is reproductively capable. She has simply been prevented from acting on the capability by the social structure she was born into. The suppression is behavioral and chemical, exerted by the queen through a combination of direct aggression, scent, and probably a pheromonal signature whose details are still being worked out in the literature. Whatever the exact mechanism, the effect is durable and almost complete. The colony has, functionally, one breeding female. The queen herself is anatomically distinct from the others in a way that is, on first encounter, faintly disturbing. She is longer, not slightly longer, visibly, measurably longer, with a body that can be a third again the length of an ordinary worker's. The added length is not in the legs or the skull or the tail. It is in the vertebral column, specifically in the lumbar region, the lower back, where the bones between the ribs and the pelvis have, over the course of her reproductive life, stretched. The vertebrae themselves have elongated. Skeletal measurements of queens taken from breeding colonies, compared with measurements of non-breeders, show lumbar vertebrae roughly 37% longer in queens. The growth is driven by reproductive activation. When a non-breeding female is removed from her colony and allowed to come into breeding condition, her lumbar vertebrae begin, within months, to lengthen. With each successive pregnancy, they lengthen further until, after roughly 8 pregnancies, the growth tapers and her adult dimensions are reached. Researchers who have written about this phenomenon describe it as a kind of puberty that is delayed by social context and then arrives, when the context allows it, as a single sustained growth event. Most queens never reach the upper limit of the growth, because most queens are killed before that by another female trying to take their place. Her behavior is no less specific. She patrols the tunnels constantly. The frequency with which she walks the colony's corridors, compared with the frequency of other adults, is high enough that the behavior shows up in any video analysis of activity inside a captive colony. When she passes a worker in a narrow stretch, the worker presses flat or rolls onto its back, motionless. When she passes a worker in a wider space, she sometimes shoves it, physically, with her body, pushing it aside or stepping directly on top of it as she continues moving. The shoving has been studied. It increases under conditions where the queen seems to be reasserting her position, and it is one of the visible mechanisms by which the reproductive suppression of the rest of the colony is maintained. A queen who shoves more, the data suggest, produces more obedient subordinates. The mating itself is a quiet affair, restricted to the queen and 1 to 3 breeding males. gestation runs roughly 70 days. Litter sizes are large for a rodent of this size, averaging 11 or 12 pups, with documented maxima reaching 27, the largest recorded litter size of any mammal. A queen can produce a new litter as soon as 80 days after the previous birth. She can do this for 30 years. She does not appear to experience menopause. Queens, still in their 4th decade of life, have been observed bearing healthy litters in captivity. Across the course of one queen's reproductive life, she may produce well over 500 offspring, the great majority of whom will live and die in the colony she rules, working for her, raising her later pups, and never breeding themselves. What this means, in evolutionary accounting, is that a colony of naked mole rats is, in the most literal genetic sense, a single extended reproductive unit. One animal's reproductive interests have, through the long action of natural selection, become the reproductive interests of every animal in the colony. The arrangement is not chosen by the workers, it is enforced by the queen, scaffolded by hormones, and underwritten by tens of millions of years of evolution. The animal that emerged from those millions of years is not exactly a colony of individuals. It is something closer to a single distributed body, with many mouths, one of which is reproductively active. A second mammal species has now been confirmed to do this, the Damaraland mole rat of southern Africa, a relative of the naked mole rat in the same general family of African subterranean rodents, but separated by tens of millions of years of independent evolution, lives in colonies organized along the same lines: a single breeding female, reproductive suppression of all others, cooperative care of the young. The two species evolved their social systems independently. That is the more remarkable fact in some ways than the original discovery. Whatever ecological pressure produced eusociality in the naked mole rat, produced it again, separately, in a related but distinct lineage. Mammalian eusociality is not, in other words, a freak accident. It is a stable evolutionary solution to a particular kind of problem, reachable by more than one road. The road in this case ran for 31 million years through the soil of East Africa. What the road produced by the time human biologists encountered it in the middle of the 20th century was an animal whose body was being shaped not only by the demands of subterranean life, but by the requirements of a society as tightly coordinated as a hive of bees, and whose physiology, beneath the surface of that social arrangement, had drifted further from the mammalian baseline than almost anyone, before Jarvis published her paper, had been willing to imagine. One of the most basic things a mammal is supposed to do is generate its own heat. This is, in fact, part of how the category was originally defined. Mammals, alongside birds, are warm-blooded, endothermic in the technical vocabulary, meaning that their body temperature is held nearly constant by the metabolic activity inside them, regardless of the temperature of the air or water around them. A laboratory mouse held in a cool room will burn calories at a higher rate, will shiver if the room is cold enough, will adjust the activity of the small brown deposits of fat across its shoulders that exist specifically to generate heat, and will maintain a body temperature of roughly 37 degrees Celsius throughout. A human will do the same, in a less dramatic way, by sweating when overheated and shivering when chilled. A polar bear will do the same in the high Arctic, by way of a deep insulation of fat and fur. The constancy of the internal temperature is one of the most reliable signatures of mammalian life. It is also, more than almost any other single physiological trait, what made the rise of mammals possible after the end of the dinosaurs. A body that could be active at any time of day and any time of year, that did not have to wait for the sun to warm it before it could move. The naked mole rat, almost alone among mammals, does not do this. Place a single isolated animal of this species in a chamber and slowly lower the air temperature, and the body temperature of the animal will track the chamber. At an ambient temperature of 30 degrees Celsius, the body temperature is roughly 30 degrees Celsius, at 25 degrees, 25, at 20, 20. The animal is not shivering. It is not adjusting its metabolic rate to defend a fixed internal set point. It is, in the strictly physiological sense of the term, behaving like a reptile. The early observations of this pattern, made by Jenny Jarvis and her colleagues in the 1970s, were so unexpected that for a long time the relevant papers carried slightly hedged language. The animal was described as behaviorally endothermic, or partly homeothermic, or imperfectly homeothermic, because the conclusion that a small pink rodent of East Africa was, in fact, cold-blooded was not the kind of conclusion that anyone wanted to commit to in print without checking it several times. The conclusion turned out to be correct. The naked mole rat is what biologists now most often call a thermoconformer, an animal whose body temperature conforms to the temperature of its environment. Some researchers still prefer the older term poikilotherm, the same word applied to fish and reptiles and amphibians. Others have argued for heterotherm on the grounds that the animal does retain some capacity for heat generation under specific conditions. The terminology has not entirely settled, and the disagreement in itself says something useful about where this animal sits in the standard mammalian taxonomy of physiology. It does not sit anywhere in particular. The vocabulary developed for ordinary mammals does not quite reach to describe it. What rescues the animal from the consequences of this arrangement is the environment it lives in and the colony it lives with. The tunnel system, 3 meters below the surface of East African savanna, holds a temperature of roughly 29 to 32 degrees Celsius year-round. This is not a coincidence. The depth of the burrow is sufficient to insulate the network from the daily and seasonal swings at the surface, and the steady output of metabolic heat from dozens of small bodies inside the tunnels keeps the internal climate stable in a narrow band that, by accident or by long evolutionary fit, corresponds almost exactly to the temperature at which the metabolism of this animal works most efficiently. The naked mole rat does not need to defend a body temperature against a hostile environment, because the environment is, in effect, holding the body temperature for it. Every member of the colony is contributing some small amount of heat to the system, and the system is contributing the heat back to every member. The single isolated animal, the one in a laboratory chamber separated from the warmth of the others, is in a kind of physiological emergency that this species was not built to handle alone. In a colony, the cold response is behavioral rather than metabolic. When the air in the tunnel drops, the animals huddle. They pile together in the nesting chamber, often in a single tangled heap of 20 or 30 or 50 bodies, and the heat of the pile, generated by the cumulative metabolic activity of the members of the pile, is conserved by the physical fact of so many bodies pressed against each other. Behavioral thermoregulation is the term used in the literature, and the term is accurate. The colony, as a unit, is warm-blooded. The individuals, as individuals, are not. This arrangement has consequences that ripple outward through the physiology of the species. A mammal that does not have to spend energy maintaining its body temperature has, for that reason, an unusually low metabolic rate. The basal metabolism of the naked mole rat is approximately 70% of what would be predicted for a rodent of its size, and under conditions of food scarcity, the animal can drop that rate further, by as much as a quarter again, into a state that conserves energy at a level rarely seen in mammals of any size. A mammal that runs cooler also produces fewer of the cellular byproducts that accumulate as a body ages. Oxidative damage, the slow chemical wear that comes from the constant burning of fuel in oxygen, scales with metabolic activity. Run the engine slower, and the engine corrodes more slowly. Researchers who work on the longevity of this animal have, for decades, pointed at the low metabolic rate as one of the factors that makes the rest of its biology possible. It is not the whole explanation. The mechanisms underneath are more specific and more strange than a simple slowing of the metabolic clock. But the slow clock is part of the picture. The cold-blooded arrangement is also part of why this animal could afford to drift, evolutionarily, in the direction it has drifted. A mammal that is rigidly committed to maintaining a 37-degree internal temperature is in many ways locked into a particular set of biochemical solutions. Enzymes, membranes, proteins, the whole molecular machinery of the body, all of it has to operate at that fixed temperature. When the temperature varies, the machinery has to be reshaped accordingly. The naked mole rat, by relaxing its commitment to a fixed internal temperature, has opened up a wider zone of biochemical possibility, and at least some of the unusual traits that show up in the rest of its physiology: the tolerance of low oxygen, the resistance to oxidative damage, the capacity for the unusual metabolic switching that this episode will return to later, may be possible in part because the animal does not have to hold its temperature steady while doing them. There is one experimental observation that captures, more cleanly than any other, how far this animal has moved from the ordinary mammalian baseline. When a naked mole rat is injected with a chemical pyrogen, a substance that triggers a fever response in any normal mammal, the animal does, in fact, raise its body temperature. The fever response, the ancient immune defense in which the body burns hotter in order to make life difficult for an infecting organism, is intact. It works, but the elevation in temperature is only sustained while the colony is present and the ambient temperature supports it. The fever, like the body temperature, is partly behavioral. The animal moves to a warmer area of the tunnel, presses against warmer bodies, and uses the structure of the colony to do what other mammals do internally. The immune response is the same. The mechanism by which the immune response is expressed has been rewired. The standard picture of a mammal, then, fails on the very first criterion in this species. The body does not generate its own heat in any robust way. The body does not defend a fixed internal temperature. The biochemistry runs at the temperature of the soil. The colony does the work that the individual cannot. And the slow, cool, low metabolic life that results, the life of an animal that runs on a smaller flame than its body size would predict, turns out to be one of the conditions under which the rest of the strangeness of this species became evolutionarily possible. Down in the corridors, in the slow heat of the nest chamber, the animals are still breathing the heavy, oxygen-poor air of the tunnel. The thermoregulation problem for them has been solved. What has not been solved, at least not by any ordinary mammalian means, is the air itself. The air in a naked mole rat tunnel is not just thin in oxygen, it is also slowly acidic because the carbon dioxide produced by every breath of every animal in the colony has nowhere to go. It accumulates. In a well-ventilated room, the carbon dioxide a person exhales drifts away, mixes into the wider atmosphere, and is gone. In a closed corridor underground with 80 bodies breathing into it across the course of a day, the gas builds up to concentrations that, in a human office, would have already cleared the room. The air in those tunnels carries carbon dioxide at levels several times higher than the atmosphere at the surface, sometimes reaching as much as 10% of the gas in the corridor by volume. When that gas dissolves into the moisture on the surface of the skin or onto the mucous membranes of the eyes and the nose and the mouth, the result is mildly acidic, a constant, gentle chemical sting against the body of any animal that has to live in it. For most mammals, a constant gentle acid is not a survivable situation. The same chemistry that makes the carbon dioxide harsh on the skin makes it harsh on the inside of the nose, on the eyes, and on the sensitive nerve endings that detect chemical irritation. A laboratory mouse placed in that atmosphere will spend a great deal of its time in distress, scratching at its face, withdrawing from contact with surfaces, and showing the signature behaviors of an animal in chemical pain. The naked mole rat does not. This was first established with some care in 2008 by a research collaboration between two groups, one in Berlin at the Max Delbruck Center for Molecular Medicine, led by a pain physiologist named Gary Lewin, and one in Chicago at the University of Illinois, led by a neuroscientist named Thomas Park. Park had been working on this animal for several years already, and one of the questions he and Lewin became interested in was, in retrospect, an obvious one. The atmosphere these animals live in should hurt them. Does it? To answer that, they used the standard set of laboratory chemicals that are known to produce pain in mammals, and they applied them in carefully controlled small doses to the skin of naked mole rats and to the skin of laboratory mice for comparison. Two substances were of particular interest. The first was dilute hydrochloric acid at a pH of 3.5, a strength comparable to the acidity of vinegar or the inside of a stomach, strong enough to produce a clear and immediate pain response in any ordinary mammal. The second was capsaicin, the active compound in chili peppers, the chemical that produces the familiar burning sensation when something spicy is eaten, or more directly, when capsaicin extract is applied to the skin. The mice responded the way mice always respond. They withdrew the paw, licked at the affected area, vocalized, and showed the small repertoire of behaviors that animal pain researchers use to score the intensity of a chemical noxious response. The naked mole rats showed nothing. Not a reduced response, not a slower response, not a partial response, nothing. A drop of dilute acid placed on the skin produced no withdrawal, no grooming, no detectable shift in behavior at all. A dose of capsaicin sufficient to cause severe burning pain in a mouse, or for that matter in the hand of a researcher unlucky enough to spill any of the working solution, produced on the skin of a naked mole rat no observable reaction. The animals continued whatever they had been doing. They moved through the experimental chamber as if nothing had been applied. They tolerated repeated doses of substances that, in any other mammal, would have produced unmistakable distress. This is the kind of result that makes a careful researcher slow down. The first explanation that occurs to anyone on reading a paper like this is that the animals must be experiencing the pain but simply not showing it, that the response is being suppressed somewhere downstream of the actual sensation, perhaps for behavioral reasons connected to colony life. The Lewin and Park collaboration was thorough enough to rule that out. They went into the spinal cord of the animal, and the peripheral nerves recorded directly from the sensory fibers that carry pain information from the skin to the central nervous system, and looked for the electrical signature of acid-induced pain. In a mouse, those sensory fibers fire vigorously when acid is applied to the skin. In a naked mole rat, they do not. The signal is not being suppressed. The signal is not being generated in the first place. What the researchers eventually traced this to is a small set of specific molecular changes in the peripheral nerves of this species. The most striking is the near-complete absence of a signaling molecule called substance P from the sensory fibers that supply the skin. Substance P is one of the central neuropeptides used by the mammalian nervous system to communicate inflammatory and chemical pain to the spinal cord. In an ordinary mammal, the fibers that respond to capsaicin and acid release this molecule into the spinal cord, where it amplifies the pain signal and helps produce the behavioral response. In the naked mole rat, those fibers do not carry the molecule. The wiring is intact. The molecular ammunition is missing. The capsaicin story is, if anything, more peculiar than the acid story. The The receptor that detects capsaicin, a protein called TRPV1, found on the surface of pain-sensing nerve cells in nearly every mammal, is fully present on the nerve cells of the naked mole rat. The receptor works. When capsaicin is applied to the cells in a laboratory dish, the receptor opens and the cell fires. The signal travels up the nerve fiber. It reaches the spinal cord. And there, because the relevant neurotransmitter is missing, the signal stops. The animal's nervous system has registered the capsaicin. The animal's behavior has not. To prove that the absence of the molecule was indeed the cause, the researchers performed an elegant test. They injected substance P directly into the spinal cord of a naked mole rat and then applied capsaicin to the skin. The animal, for the first time in the recorded history of the species in a laboratory, responded to capsaicin the way an ordinary mammal responds. It withdrew, it groomed, it showed every sign of feeling for the first time, what billions of other mammals feel every time they bite into a chili pepper. When the substance P cleared from the system, the indifference returned. There is a clean evolutionary logic to this. The atmosphere of a naked mole rat tunnel is mildly acidic in a way that, for any animal that registered acid as painful, would be a daily torment lasting an entire lifetime. The evolutionary pressure to silence that particular alarm system in an animal committed to subterranean life in densely populated colonies would have been continuous and considerable. The route to silencing it, removing a specific neuropeptide from the chain of signal transmission, is mechanistically minor in genomic terms, and it does not require dismantling the rest of the pain system. The fibers are still there, the receptors are still there, the wiring is still there. A single molecule has been quietly subtracted, and the result is an animal that does not feel the burn that defines an entire class of ordinary mammalian pain. This last point is worth stating clearly because the popular reporting on this finding has consistently mischaracterized it. The naked mole rat is not an animal that does not feel pain. It is not, in any general sense, immune to suffering. The species feels mechanical pain, pinches, pressure, sharp injuries, in the way that other mammals do, and shows the behavioral responses that go with it. A naked mole rat bitten by another naked mole rat in a colony fight will withdraw, will favor the injured side, will avoid further contact. The pain system as a whole is intact. What has been removed is a single specific channel within that system, the channel that responds to two particular kinds of chemical stress, acid and capsaicin, for which the evolutionary cost of keeping it active in this animal's environment was, evidently, more than the benefit. The same general pattern has since been found in a handful of other African mole rat species in independent forms, with different molecular routes to the same outcome. Different lineages have arrived at chemical pain insensitivity in different ways, some through the same substance P route, some through alterations in the receptor proteins themselves, some through changes in the sodium channels that carry the pain signal along the nerve fiber. The convergence is striking, whatever the ecological problem is that subterranean life poses for the mammalian pain system. The solutions are common enough that evolution has reached them more than once. The pharmacological interest in this animal, on the basis of these findings, has been considerable. Substance P, the sodium channel Nav1.7, the receptor protein TARK, these are the same molecules that have been identified in human clinical work over the last 2 decades as targets for new pain medications. The mole rat has, in effect, run an evolutionary experiment on those molecules at the scale of an entire species, and the results of the experiment have been quietly informing drug development for years. Whether any of the resulting medications will ever reach a pharmacy shelf is a separate question. The evolutionary precedent, in any case, exists. There is a way to take certain kinds of pain out of a mammalian nervous system. It has been done. It has been done in a small pink rodent of East Africa, with no design or supervision of any kind, by the slow accumulation of small genetic adjustments in the tissue of the peripheral nerves over the course of a stretch of evolutionary time long enough to swallow most of mammalian history. Down in the corridors of the colony, the animals continue to breathe the heavy carbon dioxide-rich air without distress. The acid does not bite at them. The chemical sting is, for this species, simply absent. They have lost the alarm because the alarm was no longer useful. What they have not lost, what no mammal that draws breath can afford to lose, is the deeper signal that air this thin is approaching the threshold below which the brain begins to fail. That signal too has been rewired in this animal, not silenced this time, reworked. A laboratory mouse held in an atmosphere containing 5% oxygen, roughly a quarter of the oxygen present in the air at sea level, will be dead in 15 minutes. A human climber on the summit of Everest, breathing air at roughly half the oxygen pressure of the air at sea level, is operating at the absolute upper limit of what an unaided body can tolerate. And even there, the climber is moving slowly, thinking poorly, suffering, and not staying for long. 5% oxygen is, by any conventional measure of mammalian physiology, an atmosphere that mammalian bodies cannot survive in. This is the atmosphere a naked mole rat lives in for most of its life. The exact figure varies by where in the tunnel system the animal happens to be at any given moment. Near the surface, where small amounts of air slowly exchange through the openings used for soil ejection, the oxygen concentration is higher, sometimes approaching 9%. Deep in the sleeping chambers, in the middle of the night, when the colony is piled together, breathing in the same enclosed space, the concentration can drop closer to 2 or 3. The animals do not adjust their behavior in any obvious way as they move between these zones. They eat in low oxygen, they mate in low oxygen, they give birth in low oxygen. They have, in the lifetime of any individual member of the species, never breathed the air at the surface and never will. The first careful laboratory measurements of how this animal handles low oxygen were carried out by Thomas Park's group at the University of Illinois in a series of experiments published across the 2000s. The setup was straightforward in design and unsettling in observation. The researchers placed naked mole rats and laboratory mice in sealed chambers, gradually lowered the oxygen concentration of the air inside the chambers, and recorded what happened. At 21% oxygen, ordinary atmospheric air, both species behaved normally. At 10%, the mice began to show signs of distress, while the naked mole rats continued to move around the chamber, breathing more slowly than before but otherwise unaffected. At 5%, the mice were finished, collapsed, then dead, within about a quarter of an hour. At the same 5%, the naked mole rats remained active. They explored the chamber. They groomed. They ate small pieces of food the researchers had placed inside. They did this for 5 hours. 5 hours is the figure that appears in the published papers. But the researchers eventually stopped the experiments at that point, not because the animals were failing, but because the experimenters were satisfied that the animals were going to keep going. A naked mole rat in air containing 5% oxygen is not, by any visible measure, having a bad day. The mechanisms underneath this tolerance are several, and they overlap. The lungs of this animal are small for its body size, and the airways are configured in a way that holds gas in close contact with the blood vessels for longer than is usual in mammals of comparable mass. The hemoglobin in the blood, the molecule that binds oxygen for transport, has a chemical structure that grips oxygen more tightly than the hemoglobin of a mouse or a human, meaning that at any given partial pressure of oxygen in the lungs, more of the available gas is captured rather than left to drift past. The metabolic rate of the animal is, as established earlier, low, roughly 70% of what would be predicted for a rodent of its size, and that low rate can be cut further by as much as another 25% under conditions where oxygen is scarce. The brain, in particular, shows specific adaptations that protect the neural tissue from the damage that ordinarily accompanies oxygen deprivation in mammals. The cell-to-cell calcium signaling that, in a hypoxic mouse brain, runs out of control and kills neurons within minutes is, in the naked mole rat brain, attenuated to a degree that allows the tissue to keep functioning under conditions that would destroy it in any ordinary mammal. All of these adaptations taken together explain how a naked mole rat can spend hours in an atmosphere that would kill a mouse in a quarter of an hour. They do not explain what happens when the oxygen is taken away entirely. In April of 2017, in the journal Science Park, and an international team of collaborators, Gary Lewin in Berlin, Bethany Peterson in Chicago, and a group of researchers spread across institutions in Germany, the United Kingdom, and South Africa, published a paper describing an experiment that, when read carefully, is one of the strangest things in the recent biological literature. The experiment was the next logical step after the hypoxia work. If naked mole rats can survive in 5% oxygen for hours, what happens when they are placed in 0% oxygen? Pure nitrogen, no oxygen at all. The complete and absolute absence of the gas that mammalian metabolism depends on. This is a condition called anoxia, and it is one of the most cleanly fatal environmental insults a mammal can be exposed to. A mouse in pure nitrogen loses consciousness within 30 to 40 seconds. Brain activity ceases within a few minutes. Cardiac arrest follows. By the time 5 or 10 minutes have passed, the damage to the brain is severe enough that even immediate restoration of normal air will not bring the animal back. The tissue has already begun to break down, and the animal cannot be resuscitated. A human heart attack lasting more than a few minutes produces, by the same mechanism, the brain injuries familiar from emergency medicine. The brain has very little capacity to survive without oxygen. Park and his colleagues placed naked mole rats in chambers and replaced all the air with pure nitrogen. The animals slowed. Within the first minute, their movements became sluggish. Within 2 minutes, they had collapsed. Their heart rates, monitored throughout the experiment, dropped from roughly 200 beats per minute to about 50. Their brain activity, recorded by electroencephalography, dropped to levels barely distinguishable from a flat line. They lay motionless on the floor of the chamber. By any visible measure, they were dying. They were not dying. 5 minutes passed, then 10. The animals remained motionless, heart rates suppressed, brain activity nearly absent. At 18 minutes, the researchers reintroduced normal air to the chambers. The naked mole rats stood up. They walked. They resumed grooming. Within minutes, they were behaving as though nothing had happened. There was no measurable damage to the brain, no impairment of behavior, no shortening of subsequent lifespan in the animals that were followed long-term. 18 minutes is a long time. It is roughly the duration of an episode of a television sitcom. It is longer than the time it takes most people to walk a mile. It is, in mammalian terms, an eternity of complete oxygen absence, more than 30 times longer than any other mammal of comparable size can tolerate without irreversible brain injury. The naked mole rats walked through it the way an ordinary animal might walk through a brief power outage. The question that the experimental result immediately raised was not whether the animal could do this. The experiment had clearly demonstrated that it could, but how? Mammalian metabolism, the biochemistry that runs every cell in every mammal alive, requires oxygen. the energy currency that powers muscle contraction, nerve firing, the work of every organ. The molecule called adenosine triphosphate, ATP, is produced primarily through a chemical pathway called oxidative phosphorylation, which runs inside the mitochondria of every cell and which cannot function without oxygen at the end of the chain. Cut off the oxygen and the production of ATP collapses. The cells, deprived of their energy source, fail. In short order, the tissues die. There is a backup. In the absence of oxygen, mammalian cells can produce ATP through a different pathway, anaerobic glycolysis, which extracts a smaller amount of energy from glucose without requiring oxygen at the end. Sprint athletes use this pathway when their muscles outrun the available oxygen supply during a 100-meter dash. The pathway is fast, but it is inefficient. It produces lactic acid as a waste product, and it has a built-in molecular brake. After a short period of operation, an enzyme called phosphofructokinase, which sits at one of the early steps of the pathway, shuts itself down in response to feedback signals from the system. The pathway closes. In an ordinary mammal, anaerobic glycolysis can keep the body running for, at most, a few minutes. After that, the brake engages, the pathway stops, and the cells have no remaining options. 18 minutes of anoxia in a mammal should not be survivable by any of the routes known to mammalian biochemistry. When Park and his colleagues analyzed the blood and tissues of naked mole rats that had been exposed to anoxia, they found something that, on first inspection, did not look like a mammalian metabolic profile at all. The animals were running on a sugar that mammalian metabolism, under ordinary conditions, almost never uses. Glucose is the sugar that runs the mammalian body. It is the molecule that arrives in the bloodstream after a meal, that is delivered to every tissue, that is broken down inside every cell to produce the small unit of chemical energy that powers everything from a heartbeat to a thought. The entire metabolic infrastructure of mammals, the enzymes, the transport proteins, the regulatory feedback loops, the way insulin signals across the body, is built around glucose. When biologists talk about blood sugar, they are talking about glucose. When a doctor measures blood sugar, the measurement is of glucose. The mammalian body, in any practical sense, runs on this one molecule. There is a closely related molecule, almost identical in chemical structure, called fructose. The two sugars share the same molecular formula. They differ in the arrangement of their atoms, a small structural shift that, in chemical terms, looks almost trivial. Fructose is the sweetness in fruit, the sweetness in honey, and one half of the structure of ordinary table sugar, which is a compound called sucrose, made of one glucose molecule joined to one fructose molecule. In dietary terms, mammals encounter fructose all the time. The body, however, handles it differently from glucose. Most fructose absorbed from food is processed in the liver, broken down into other molecules, and rerouted into the wider metabolic system. It is not used directly in the cells of most tissues as a fuel. The receptors and enzymes required for direct fructose-based energy generation are, in most mammals, either absent from most tissues or present at levels too low to do meaningful work. This is the point at which the naked mole rat does something that no other mammal so far studied is known to do. When Park and his colleagues analyzed the tissues of animals that had been exposed to anoxia, they found dramatically elevated levels of fructose throughout the body. The concentration of fructose in the blood of an anoxic naked mole rat reached more than 240 micromoles per liter, several times the resting level in the same animal under normal conditions, and several orders of magnitude higher than the fructose concentration ever measured in the blood of a mouse under any conditions whatsoever. The fructose was not coming from food, the animals had not eaten anything unusual. The fructose was being produced by the body somewhere in response to the loss of oxygen. Even stranger, the fructose was being used. Not stored, not shuttled off to the liver and discarded, but actively transported into the cells of metabolically demanding tissues, the heart, the brain, the kidneys, and broken down there to produce energy. The transport protein that carries fructose across cell membranes, called GLUT5, was being expressed at high levels across these tissues. The enzyme that performs the first chemical step of fructose breakdown, called ketohexokinase, was also present at unusually high levels. The molecular infrastructure for direct fructose metabolism, missing or undeveloped in most mammalian tissues, was fully built in this animal. What that infrastructure allowed the naked mole rat to do, biochemically, is the central trick. The reason mammals cannot survive long without oxygen is, at root, a problem with one specific enzyme in the glucose breakdown pathway. The enzyme is the phosphofructokinase mentioned earlier, the molecular brake that shuts down anaerobic glycolysis after a few minutes of running. The brake is not a flaw in the system. It is a regulatory feature, evolved to prevent the cell from poisoning itself with lactic acid and from depleting its limited supply of glucose under prolonged stress. The feature, however, has a price. Once the brake engages, the pathway closes, and the cell has no remaining way to generate energy in the absence of oxygen. The cell fails, the tissue dies. Fructose, when it enters the same general pathway, enters it through a different door. The early steps of fructose breakdown bypass the phosphofructokinase brake entirely. The molecule is processed by a different set of enzymes, ketohexokinase, then aldolase B, then a few more, and reaches the same downstream chemistry that ordinary glucose breakdown reaches, but it arrives there without ever passing through the regulatory choke point that would normally shut the system down. The brake is invisible to a cell running on fructose. The pathway remains open. Energy production continues. This is what was happening biochemically in the bodies of the 18-minute anoxic naked mole rats. Their cells had stopped processing glucose because the glucose pathway had hit its brake. Instead, their tissues had switched to fructose. The fructose was being transported into the heart and the brain by the GLUT5 protein, processed by ketohexokinase, and converted into the same end product, lactate, that anaerobic glycolysis produces, but by a route that the regulatory machinery of the cell does not know how to shut down. The naked mole rats were, in effect, running on a different sugar through a different door, and the door did not close. There is one detail of this story that is worth saying out loud because it appears in almost none of the popular reporting on the finding. Fructose-driven glycolysis, of the kind that the naked mole rat does in anoxia, is the standard way that many plants generate energy. Plants, lacking the kind of mobile circulatory system that delivers oxygen-rich blood to every tissue, frequently run sections of their metabolism on fructose-based pathways that bypass the kinds of regulatory feedback that mammalian glucose metabolism depends on. The biochemistry that the naked mole rat deploys during anoxia is not, in its essential outlines, a new invention by mammalian biology. It is the appropriation, by an evolutionary route still being worked out, of a metabolic strategy that the rest of the kingdoms of life, plants, certain fungi, many microorganisms, have used for hundreds of millions of years. When the popular press headlines for this paper read, "Naked mole rats turn into plants when oxygen is low," the headlines were being more accurate than is usual for popular press headlines on biology stories. This does not mean the naked mole rat is a plant in any sense that needs taking seriously. The animal is still a mammal in its developmental history, its anatomy, its reproductive biology, and the great majority of its everyday physiology. What the animal has done is something narrower and, in its own way, more interesting. It has reached into the toolkit of biochemical strategies that exist on Earth, picked up a tool that the rest of the mammalian lineage left untouched, and deployed it inside its own cells. The tool was lying there, available, used by other lineages for other purposes. The naked mole rat is the only mammal known to have picked it up. How exactly this happened is still under investigation. The genetic changes that produce the high expression of GLUT5 and ketohexokinase across the heart, brain, and kidneys of this species are described in the supplementary material of the 2017 paper, and they involve modifications to the regulatory regions of the genes rather than radical changes to the protein coding sequences themselves. The proteins are not, by and large, fundamentally different. They are simply being made, in this species, in tissues where in other mammals they would not be made, or would be made at much lower levels. The trick is in the where and the when, not in the what. A small set of regulatory shifts applied across the body opens up a metabolic capability that was, until those shifts occurred, unavailable to mammalian biochemistry. What this implies, in the broader picture of biology, is that the boundary between mammalian and plant metabolism, between an animal that needs oxygen and a kind of life that does not, or that does not in the same way, is less rigid than the categorical division suggests. Living things share, at the level of the chemistry, an enormous common toolkit. The categories are real, but they are not walls. They are, in many cases, particular subsets of the toolkit that one lineage happens to have settled on. When evolutionary pressure is strong enough, a lineage can occasionally reach across into a subset that other lineages have been using all along and bring back a tool that the original assumptions of the category did not predict. That is what has happened, on a small but cleanly demonstrated scale, in this animal. A mammal has reached into the metabolism of plants and brought back the capacity to survive without oxygen. The reach is partial, the duration is finite, the mechanism is specific, but the reach has happened. It has happened in a body that fits in the palm of a human hand and lives 3 meters under the dry soil of East Africa, and has been quietly running this evolutionary experiment for several million years before anyone in a laboratory ever thought to ask whether such a thing was possible. The asking, when it finally came, was done by a man in a small laboratory in Chicago. The experiment that followed produced on a winter afternoon in 2016 a scene that the people in the room have not entirely stopped talking about. The room was small, a laboratory on a quiet floor of a building on the west side of Chicago, 2 doors down from a coffee machine that nobody had cleaned in a while, a row of computers, a glass-fronted environmental chamber sitting on a stainless steel bench. The chamber was sealed against the surrounding air, fed by precision gas lines from cylinders along the wall, and instrumented with sensors that measured the oxygen and carbon dioxide concentrations inside it down to a fraction of a percent. The fluorescent lights overhead had been dimmed slightly because the animal in the chamber was used to working in the dark. Inside the chamber, on a small bed of soft bedding, was a single adult naked mole rat. She had been part of a captive colony for most of her life, and she had no particular reason on this afternoon to expect anything unusual. The researchers running the experiment, Thomas Park and 2 of his graduate students with a remote feed open to Gary Lewin's lab in Berlin, had spent months developing the protocol. Anoxia experiments in mammals are not the kind of thing that anyone runs casually. The protocols are carefully approved. The recovery conditions are precisely specified. The equipment is checked and rechecked, and the people in the room have already, in their heads, walked through what they expect to see. What they were expecting to see was the animal slow down, lose consciousness within a few minutes, and then, if everything worked, recover when the oxygen was restored at the predetermined point. They were expecting, in short, a controlled survival experiment. What they got when they ran the experiment was something stranger than that, and the strangeness was visible from the first 30 seconds. The gas flow was started just before 2 o'clock in the afternoon. Pure nitrogen began to displace the air inside the chamber. The oxygen sensor, which had been reading the laboratory's ambient 21%, began to drop. 15%, 10, 5. Within roughly 2 minutes, the reading was below 1% and still falling. The animal in the chamber, who had been moving slowly around her bedding, paused. Her breathing slowed. Her movements became less coordinated. Within another minute, she had lowered herself onto her side and stopped moving altogether. On the monitor at the bench, the heart rate trace showed 200 beats per minute, dropping toward 100, then toward 80, then toward 50. In a mouse, this is the point of no return. In a mouse, the next 40 seconds would be the end of the experiment, regardless of what the experimenters did with the gas valves. The brain tissue would have begun the irreversible cascade that follows oxygen loss, the cells failing one after another in the precise sequence that emergency medicine has documented in human cardiac arrest patients for decades. The animal would be, in the technical and irreversible sense, dying. The naked mole rat lay on the bedding without moving. The oxygen reading reached 0. The chamber held. The minutes began to pass. At 5 minutes of anoxia, the animal's heart rate was around 50 beats per minute, holding steady. Her breathing had slowed to a rate so low that the chest movements were difficult to see without staring. The brain activity trace monitored by an external device was flatter than the trace of a healthy sleeping animal, close to silent, but not entirely silent. The instrumentation was, in some senses, picking up the signal of a deeply suppressed but still functioning nervous system. At 10 minutes, nothing had changed. The animal had not deteriorated. The heart rate had not dropped further. The brain trace was no flatter than it had been at 5 minutes. The body in the chamber looked, by any superficial measure, like the body of a small dead animal, but the instruments were quietly reporting that the situation was stable. Whatever was happening inside the cells of the heart and the brain and the kidneys, it was a kind of stable that the laboratory's reference textbooks did not describe. At 15 minutes, the researchers exchanged a look. At 18 minutes, by the agreed-upon protocol, Park opened the gas line for normal air. The oxygen concentration began to climb back toward atmospheric levels. The chamber refilled slowly with the same air it had started with at the beginning of the afternoon. The instruments tracked the recovery. Within 30 seconds of the oxygen being restored, the brain activity trace lifted off the floor and began to climb. Within a minute, the heart rate had moved from 50 back toward 80. Within 2 minutes, the breathing rate had picked up to something approaching normal. At 4 minutes, the animal moved a leg. At 5 minutes, she lifted her head. By the 10-minute mark of recovery, the naked mole rat had stood up. She walked around the chamber. She investigated the bedding. She nosed at the food that had been left for her. By every behavioral measure available to the people watching, she was the same animal she had been before the experiment began. No staggering, no neurological tremor, no failure of coordination, no apparent disorientation. The protocol called for an extended observation period, with neurological and behavioral testing over the following days and weeks. The testing eventually showed what the first 10 minutes had already suggested. The animal was unharmed. This is the experiment that, when it was published in the journal Science in April of 2017, produced one of the most quietly disorienting paragraphs in the recent biological literature. The paragraph reads, in part that the naked mole rat survives total anoxia by switching to anaerobic metabolism fueled by fructose, which is actively accumulated and metabolized to lactate in the brain. The paragraph reads like ordinary metabolic biochemistry. It is also, when read with the implications followed all the way through, a description of a mammal becoming, for the 18 minutes between the closing of the oxygen valve and the opening of the air line, an organism that the textbooks of mammalian biology have no name for. Here is the moment to stop and look at what this means. A mammal is, by every standard definition, an oxygen-dependent organism. The entire architecture of the mammalian body, the four-chambered heart, the lungs, the elaborate hemoglobin chemistry of the blood, the energy-generating machinery of the mitochondria inside every cell, is built around a single underlying assumption. The body needs oxygen continuously to function. Remove the oxygen, and the body fails within minutes. Of all the categorical features that biologists use to define what a mammal is, this is one of the most basic. It is more basic than warm-bloodedness. It is more basic than fur. It is more basic than live birth or maternal milk. The presence of an obligate oxygen requirement is, in essence, the underlying physical reality on which most of the rest of mammalian biology has been built. The naked mole rat does not fully share that reality. Not always. Not under all conditions. Under the specific conditions of complete oxygen absence, this animal performs a biochemical maneuver that mammals are not supposed to be able to perform, and, having performed it, walks out the other side with no measurable damage and resumes its life as though nothing had happened. The categorical feature is bent, not broken, not entirely, but bent enough that the body, for those 18 minutes, is running on a chemistry that biologists associate with the rest of the kingdoms of life, not with this one. There is no way to say this without sounding more dramatic than the science wants to be. The naked mole rat is still, genealogically, a mammal. Its ancestors were mammals. Its descendants will be mammals. The category, as a phylogenetic claim, still applies to it. But the category, as a physiological description, as a set of constraints and capabilities that the body must possess, has loosened in this lineage to the point where one of its most basic assumptions is, under stress, no longer reliable. This is the moment in the story of this animal where the small, accumulating strangenesses of the previous chapters come together into a single observation. Thermoconformity by itself is unusual, but not unprecedented. Some other mammals approach it. Chemical pain insensitivity by itself is striking, but has analogues elsewhere in the rodent world. Hypoxia tolerance of the kind the species displays under 5% oxygen for hours, while extreme, can be partially explained by adaptations that other diving and burrowing mammals show in milder forms. 18 minutes of complete oxygen absence, recovered without injury, fueled by a sugar that the mammalian body is not supposed to use as a primary fuel. This is not on the same scale as the others. This is a different kind of departure. What it suggests, when laid alongside everything else that has been said about this animal across the preceding chapters, is that the body of the naked mole rat is not a slight variation on the mammalian theme. It is something further from the standard model than the popular reporting tends to convey, and possibly something further from the standard model than the scientific literature has yet entirely come to terms with. The animal that lives under the dry soil of East Africa is, by every reasonable phylogenetic measure, a mammal. By the measure of what its body can do, it is at the outer edge of what a mammal can be, and on at least one specific axis, the relationship between body and oxygen. It has stepped past that edge into territory the category was not designed to include. This is, in one form of words, why the laboratories of the world keep coming back to this species. The question being asked of the naked mole rat is not really or not only what kind of mammal it is. The question being asked is what the category of mammal can be made to do under sufficient evolutionary pressure in sufficient time. The answer that this small pink animal has been providing in the careful experiments of Chicago and Berlin and Rochester and Shanghai across the last 20 years is that the category can be made to do considerably more than the textbooks suggest. The walls of the category in this lineage have moved. Back in the chamber, by the time the afternoon was over, the animal had been returned to her home colony. She rejoined the others. She slept that night in a pile of warm bodies, in the same 80% humidity and slightly elevated carbon dioxide that she had been born into. There is no indication that she had noticed anything unusual about her day. The discovery did not begin with a hypothesis. It began with a vacuum pump that would not stop clogging. In a building on the southern edge of the University of Rochester campus in upstate New York, a husband and wife pair of biologists named Vera Gorbanova and Andrei Seluanov were running a comparative biology laboratory. Both had emigrated from the former Soviet Union, both had trained in the United States in the early 2000s, and both had become interested in a question that, at the time, was unfashionable. The standard view of cancer biology throughout most of the 20th century was that the disease was largely a matter of bad luck and accumulated genetic damage, the inevitable downstream consequence of a long-lived body's cells dividing too many times. The standard view, in other words, treated cancer as something close to a universal property of complex multicellular life. Gorbanova and Seluanov found this view incomplete. They were interested in animals that, on the evidence, did not seem to get cancer. By the early 2000s, the naked mole rat had become one of the more obvious species to ask the question about. Rochelle Buffenstein's colonies, by then more than 2 decades old, had produced a striking observation. Across thousands of animals followed across decades of laboratory care. Animals that, on the actuarial expectations of their body size, should have shown cancer rates comparable to the laboratory mouse, where roughly 95% of individuals develop tumors before the end of their lives. The naked mole rats were producing almost no tumors at all. The published record, by the mid-2000s, contained 0 confirmed cases of cancer in this species. 0 across more than 3,000 animals examined at autopsy. The figure was the kind of figure that, in cancer biology, demands an explanation. Gorbanova and Seluanov went looking for one. The standard way to look for the cellular basis of cancer resistance is to take the cells of the animal in question out of the body and grow them in tissue culture in the flat glass-bottomed plastic dishes that have been the standard equipment of cell biology since the 1950s. The cells settle on the bottom of the dish, attach themselves to the surface, and begin to divide. Under ordinary conditions, mammalian cells will keep dividing until they fill the bottom of the dish. They will spread, touch each other, run into each other on the surface, and eventually cover the available area with a single dense sheet. At that point, in healthy cells, division slows and stops in a phenomenon called contact inhibition. Cancer cells, by contrast, ignore the signal. They keep dividing, pile on top of each other, and grow into the kinds of disorganized masses that, in a living body, become tumors. The cells of the naked mole rat, when grown in this standard setup, behaved unusually well. They divided slowly. They paid careful attention to contact inhibition, stopping when they encountered their neighbors. They did not pile up. They did not grow into transformed colonies, even when the researchers tried to push them into doing so, using the standard laboratory techniques that reliably transform mouse cells into cancerous lines within weeks. The naked mole rat cells held their order. They did not become malignant. This was, in itself, an interesting finding. What turned the finding into a mechanistic story was the vacuum pump. The laboratory was using vacuum suction to remove the growth medium, the liquid that bathes the cells in their dish, during routine media changes. With ordinary cells from ordinary species, this is a simple operation. With the cells of the naked mole rat, the operation kept getting in the way of itself. The medium being suctioned off was thick. It was viscous. It clogged the suction lines, gummed up the connections, and slowed the technicians down to the point where the routine task was becoming a bottleneck for the work. The technicians, after a while, mentioned this to the principal investigators. Gorbunova and Selovanov looked into it. What they found in the growth medium of the naked mole rat cells was an unusually large amount of a substance called hyaluronic acid. Hyaluronic acid, also known as hyaluronic, is not, by itself, an unusual molecule. It is a long, repeating chain of two sugar units, glucuronic acid and N-acetylglucosamine, alternating along a polymer backbone. It is present in the connective tissues of every vertebrate, from fish through mammals, and it is one of the central components of the extracellular matrix, the chemical scaffolding between cells that gives tissues their structural integrity. It is the slippery substance in the synovial fluid that lubricates joints. It is part of what gives skin its elasticity. It is, in cosmetic dermatology, the active ingredient in many of the injectable fillers used to soften facial lines. The molecule itself is unremarkable. What was unusual about the hyaluronic acid in the dish of naked mole rat cells was its size. Hyaluronic acid exists in different sizes depending on how long the polymer chain has been allowed to grow before it is released. The size is measured in molecular weight, expressed in units called daltons, or more conveniently, in megadaltons, millions of daltons. In human tissue culture, the hyaluronan secreted by cells typically falls in the range of 1 to 2 megadaltons. In mouse tissue culture, the figure is similar, perhaps a little higher. In the dishes of naked mole rat cells, the figure was between 6 and 12 megadaltons. The molecules were several times longer than the equivalent molecules secreted by any other mammalian cell line that anyone had previously examined. They were so long and so abundant that the medium they were dissolved in had become, in a literal sense, sticky. This was the finding that turned the cancer-resistant story from a puzzle into a mechanism. Long-chain, high molecular weight hyaluronan, the form the naked mole rat cells were producing, has biochemical properties that are different from those of the shorter chains found in most other species. The short chains, the ones found in mice and humans, are not strongly anti-inflammatory. In some contexts, they actively promote inflammation. The long chains do the opposite. They suppress inflammation. They also do something more specific, which is to engage receptors on the surface of cells in a way that strengthens contact inhibition. The same signaling pathway that, in cancer, fails. Cells embedded in a matrix rich in long-chain hyaluronan are unusually responsive to their neighbors, unusually willing to stop dividing when they touch, and unusually resistant to the kinds of genetic changes that, in cells embedded in shorter-chain matrices, set off the cascade toward malignancy. The mechanism was elegant in a way that biological mechanisms rarely are. A single class of large molecule secreted into the space between cells across many tissues of the body, holding the population of cells in a chemical condition under which the runaway division of cancer is much less likely to begin in the first place. The Gorbunova-Selovanov group, with their collaborator Xiao Tian, published the finding in Nature in 2013. The paper identified the responsible enzyme, a protein called HAS2, hyaluronan synthase 2, found in most mammals but unusually active in the naked mole rat. and demonstrated that when the long-chain hyaluronan was removed from naked mole rat cells, the cells became newly vulnerable to cancerous transformation. The mechanism was not the whole story of cancer resistance in this animal, but it was the first piece of the story to be cleanly explained. The popular reporting on the paper in the years that followed did what popular reporting on biology results often does. It compressed the finding. It rounded the edges. It produced, in news stories and viral video captions and social media posts, the phrase that has stuck to this animal in public memory ever since: "The naked mole rat is immune to cancer." The phrase is not quite right. In 2016, two separate veterinary pathology groups published case reports that, between them, documented something that the popular framing had implied was impossible. A team at the National Zoo and several associated institutions reported 2 cases of cancer in zoo-housed naked mole rats, one in an adenocarcinoma of likely mammary or salivary origin, the other in a tumor of the gastric mucosa. A team at Disney's Animal Kingdom reported separately 4 confirmed cases of cancer and 1 presumptive case across a colony of 37 autopsied animals at their facility, including a metastasizing hepatocellular carcinoma, a nephroblastoma, a multicentric lymphosarcoma, and a presumptive Barrett's-like esophageal adenocarcinoma. By the time these reports were absorbed into the wider scientific literature, the position had been recalibrated. The current count, across all the laboratory and zoo colonies of this species that have been carefully autopsied, sits at roughly 5 confirmed cases of cancer across more than 3,000 examined animals. 5 cases out of 3,000 is not zero, and the word immune does not capture what the data actually show. The accurate phrase, which the careful scientific literature has used consistently since the 2016 case reports were published, is remarkably resistant. The naked mole rat develops cancer at a rate that, by comparison with mice or humans, approaches 0, but does not quite reach it. The mechanism that produces this near resistance, the high molecular weight hyaluronan, the strengthened contact inhibition, the long list of additional cellular and immunological factors that have been identified in the years since the 2013 paper, is not perfect. It is, however, the best example of evolved cancer resistance currently known in any mammal, and the cellular mechanisms underneath it have become, over the past decade, one of the more active areas of translational cancer research. The calibration matters because the distance between immune and remarkably resistant is the distance between a slogan and a research program. A slogan invites people to expect a miracle. A research program asks what specifically is happening in this body that is not happening in others, and how much of it can be transferred. The careful version of the story, the version where the resistance is profound but not absolute, the mechanism is multiple but not magical, and the case reports are honestly counted, is the version that has, in the years since the 2013 paper, started to produce actionable results in laboratories beyond Rochester. One of those laboratories took the most central piece of the mechanism, the hyaluronan-producing enzyme itself, and asked a question that 15 years earlier, almost no one in cancer biology would have thought worth asking out loud. The question was whether the trick could be borrowed. The idea was simple enough to state in a single sentence. If the cells of the naked mole rat are resistant to cancer because they secrete an unusually long form of hyaluronan, and if the enzyme that produces that long form is the naked mole rat version of a protein called HAS2, then it should be possible in principle to take the gene that codes for the naked mole rat HAS2, insert it into the genome of a different species, and see whether the cells of that second species begin to do what the cells of the naked mole rat do. In practice, this kind of experiment is not simple at all. transferring a gene from one mammal to another, having that gene expressed in the right tissues at the right levels, having the resulting protein perform its function in a body that did not evolve alongside it, and then following the recipient animals across enough of their lifespan to see whether anything has actually changed. This is the kind of project that takes years, requires substantial funding, and depends on the patient maintenance of careful breeding colonies under controlled laboratory conditions. The Gorbunova and Seliwanov group spent most of a decade after the 2013 Nature paper preparing exactly that kind of experiment. The work was published in August of 2023 in Nature again, with Zhihui Zhang as the lead author and a long list of collaborators including Xiao Tian, who had been the lead author of the original 2013 paper, alongside contributions from groups at Harvard, at the Brigham and Women's Hospital, and at the University of California, Los Angeles. The team had inserted the naked mole rat version of the HAS2 gene into the genome of a laboratory mouse, generating a transgenic line they called NMR-HAS2, naked mole rat HAS2, and they had followed the resulting animals across their full lifespan, comparing them with ordinary mice of the same genetic background that did not carry the inserted gene. The first thing the transgenic mice did was produce hyaluronan, not in the small amounts characteristic of ordinary mouse tissue, but in elevated amounts across multiple organs: the skin, the gut, the kidneys, the heart, the liver. The hyaluronan they produced was also longer than the hyaluronan of ordinary mice, shifted toward the high molecular weight range that characterizes the naked mole rat. The biochemical part of the experiment had worked. The mouse cells were, in this respect, doing something that mouse cells do not ordinarily do. What happened next was the part that mattered. The transgenic mice developed fewer spontaneous tumors than the control mice. When the researchers deliberately tried to induce skin cancer in the two groups using a standard chemical carcinogenesis protocol with a compound called DMBA, followed by an inflammatory promoter called TPA, a combination that reliably produces skin tumors in ordinary mice, the transgenic line showed significantly fewer tumors than the controls. The reduction in spontaneous cancer incidence in the older transgenic animals, when the numbers were finally tallied, came in at roughly 34%. The health span effects went beyond cancer. Across the transgenic line, levels of low-grade inflammation in multiple tissues were lower than in the controls. The intestinal barrier, the layer of cells that separates the contents of the gut from the rest of the body and that deteriorates with age in most mammals, held its integrity better in the transgenic mice. The composition of the gut microbiome, measured in old animals, looked more like that of younger animals than the microbiome of the age-matched controls. The transcriptional profile of multiple tissues, the pattern of which genes are switched on at what levels across the body, shifted in the transgenic line in the direction of the profiles seen in longer-lived mammalian species, and the animals lived longer. The median lifespan of the transgenic mice was approximately 4.5% longer than that of the controls. The maximum lifespan, the age at which the longest-lived 10% of the population died, was approximately 12% longer. These are not transformative numbers. They are not the kind of figures that would, if extrapolated naively to a human population, add years of healthy life to most people. They are, however, numbers that are statistically robust, reproducible across two independent transgenic founder lines, and consistent with the mechanism that had been proposed in the original 2013 paper. A piece of the naked mole rat's cancer resistance machinery had been ported into a different species. The piece had worked. This is a result that needs to be held carefully on both sides. On one side, the result is genuinely important. It is the first demonstration in a controlled experiment on a mammalian species other than the naked mole rat that a specific evolved mechanism of cancer resistance can be transferred across species and produce the expected effects in the new host. The transfer worked. The mechanism is portable. The implication in the broader picture of cancer biology and aging research is that the unusual longevity traits of long-lived mammals are not necessarily locked inside the species that evolved them. At least some of those traits, identified at the molecular level and packaged as discrete genetic interventions, can be moved. On the other side, the result is a single experiment in mice. The transition from a mouse experiment to a human intervention is the long and well-known difficulty of medical research. The history of biomedicine is littered with promising mouse experiments that did not translate to human benefit for reasons ranging from species-specific differences in immune response to differences in the way the same molecule is metabolized in different bodies to differences in the underlying biology of the disease being targeted. A transgenic mouse with elevated long-chain hyaluronan is not the same thing as a human treatment based on long-chain hyaluronan, and the path from one to the other is, at present, almost entirely speculative. There is also a question, which the 2023 paper itself raised, about which features of the naked mole rat's cancer resistance are captured by the HAS2 transfer and which are not. The hyaluronan-based mechanism is the most cleanly characterized piece of the cancer resistance puzzle in this species, but it is almost certainly not the only piece. Subsequent work has identified additional mechanisms. An unusually active form of one of the standard tumor suppressor genes, called P16INK4A, that triggers cell cycle arrest at lower cell densities than in most mammals, a dampened inflammatory response to chemical carcinogenesis, a particular configuration of ribosome quality control that catches translation errors more efficiently than in mouse cells. Differences in the way certain DNA damage signaling pathways are wired, differences in the structure and abundance of glutathione, the cellular antioxidant. The cancer resistance of the naked mole rat is, in the picture that has emerged across the last decade, a stacked defense rather than a single mechanism. The HAS2 piece is the first one that has been successfully ported. Whether others can be ported and what happens when multiple mechanisms are transferred together is the subject of ongoing work in several laboratories. The 2023 paper was followed in October of 2025 by another publication in Science that opened a different door into the same general question. A research group at Tongji University in Shanghai, led by Ying Jiang and Jiong Mao, reported a finding that none of the earlier work on this animal had predicted. They had identified a small set of 4 amino acid substitutions in a specific position of a protein called CGAS that distinguish the naked mole rat version of the protein from the versions found in mice and humans. The protein in question, cyclic GMP-AMP synthase, to give it its full name, sits at the center of one of the cell's basic alarm systems, detecting fragments of foreign or damaged DNA and triggering an inflammatory response when it finds them. In mice and humans, this protein has a second function, less widely appreciated, which is to suppress one of the major mechanisms of DNA repair. Cells that need to repair damaged DNA are, in effect, working against the action of their own CGAS. In the naked mole rat, the 4 amino acid changes flip the balance. The naked mole rat version of CGAS does not suppress DNA repair the way the mouse or human version does. Instead, it allows the repair machinery to function more freely, and the resulting cellular state is one of stronger and faster repair of the kinds of DNA breaks that, in ordinary mammals, accumulate as the body ages. The Tongji group went on to insert the naked mole rat version of CGAS into the cells of mice and into the cells of fruit flies, and reported that both species, after the substitution, showed measurably improved DNA repair, fewer signs of cellular aging, and, in the case of the fruit flies, extended lifespan. This is the most recent piece of the puzzle to have been added to the picture, and it is, by some distance, the most evolutionarily peculiar. 4 amino acid changes in a single protein, accumulated in this lineage at some point during its 31 million year separation from the rest of the mole rat family, are sufficient to convert a protein that normally hinders cellular DNA repair into a protein that supports it. The change is structurally small, 4 amino acid substitutions out of a protein of several hundred residues. The functional consequence is large. The implication, when laid alongside the HAS2 story, is that the naked mole rat's resistance to age-associated disease is being produced by a series of small, specific molecular shifts of this kind, distributed across the genome, each of them individually subtle but cumulatively powerful enough to produce the longevity and the cancer resistance that the species displays. The 2025 result is too recent to have produced its own borrowing experiment in the way the 2013 paper eventually produced the 2023 one. But the work has begun. Several laboratories, including the original Tongji group, and at least one group in the United States, are now examining whether the naked mole rat CGAS variant can be transferred into the cells of other mammals, and whether the resulting cells show the same improved DNA repair. The translational question is open. The mechanism is identified, the experimental tools exist, and the timeline that produced the 2023 HAS2 transgenic mice, roughly 10 years from mechanism identification to whole animal transfer, suggests that an answer to the CGAS portability question will not arrive overnight, but will in some form arrive. What the past decade has produced on the cancer and aging side of the naked mole rat literature is a slow accumulation of mechanism. The picture is no longer one of a mysterious animal that does not get cancer, sitting in a laboratory and refusing to explain itself. The picture is a list of specific molecular shifts, hyaluronan length, P16 activation thresholds, inflammatory response dampening, ribosome quality control, DNA repair regulation through CGAS, each of which has been identified, each of which has been characterized at the level of the relevant genes and proteins, and at least one of which has now been demonstrated to produce measurable benefits when transferred into a different mammalian species. The list is not complete. Several of the items on it are still partially understood. But the list exists in a way that in 2005 it did not. The animal that has been producing these findings is, on its own time, doing what it has always done. The colonies in Chicago and Rochester and San Francisco and Cambridge and Berlin and Tokyo continue their daily routines, breathe their heavy carbon dioxide-rich air, digest their tubers, raise their pups, suppress one another's reproductive cycles, and live year after year in the strange biological condition that the previous chapters have tried to describe. Whatever the laboratories of the world have learned from them, the animals themselves have not changed at all. The questions in the laboratories have. And one question, which the cancer story and the CGAS story and the HAS2 transgenic story all touch from different angles, remains the largest one. The animal in the colony, at 20 years old, looks and behaves and tests by every available measure, exactly like the same animal looked and behaved and tested at 5 years old. Across 2 decades of life, the body of this species seems, in some deep biological sense, not to be aging, what it might mean for a mammal not to age, what the actual measured trajectory of mortality in this species looks like, and how that trajectory compares with the law that has governed the aging of every other mammal ever studied, is the next part of the story. In 1825, in a paper presented to the Royal Society of London, a British actuary and amateur mathematician named Benjamin Gompertz published one of the most successful descriptive formulas in the history of biology. The paper was not, on its surface, a paper in biology. It was a paper about the mathematics of mortality, written by a man whose professional life was spent calculating the rates at which insurance customers were likely to die. Gompertz had noticed something in the actuarial tables. The risk of dying, for any given person, was not constant across their lifespan. It rose with age. More than that, it rose with age in a very specific way. It rose exponentially. The chance that an adult would die in the next year roughly doubled every 8 years or so, regardless of the absolute age of the person being asked about. A 40-year-old was twice as likely to die in the coming year as a 32-year-old. A 60-year-old was twice as likely to die as a 52-year-old. The doubling continued, in roughly the same way, all the way to the upper limits of the human lifespan. Gompertz wrote down the equation that described this pattern. The formula had a striking property. It worked. It worked for British insurance customers in the 1820s, and it worked when applied to the actuarial records of other populations, and as the years passed and biology developed into a quantitative science, it kept working. It worked for almost every mammal that anyone tried it on: laboratory mice, rats, dogs, horses, sheep, cats, primates of various species, the wild populations of mammals that ecologists tracked across decades, across an enormous range of species and conditions. The same basic pattern appeared. After a certain point in adult life, the risk of dying began to climb exponentially with age. The doubling time varied from species to species, but the underlying shape of the curve did not. Mammals aged. The aging followed Gompertz's law. The pattern was so consistent across so many decades of measurement that for most of the 20th century it was treated without much fuss as one of the basic biological regularities of mammalian life. In 2018, in a paper published in the journal eLife, a team of researchers from Calico Life Sciences in South San Francisco, the longevity research company set up by Google's parent organization a few years earlier, and from the laboratory of Rochelle Buffenstein, who had by that point moved her naked mole rat colonies from Texas to the Calico facility, reported that this universal pattern was, in one specific mammal, not what was happening at all. The paper was a piece of careful demographic analysis. The lead author, J. Graham Ruby, together with Megan Smith and Buffenstein herself, had assembled a dataset of more than 3,000 naked mole rats whose dates of birth and dates of death had been precisely recorded across more than 3 decades of laboratory care. They had looked at the daily probability of death as a function of age. They had asked the simple question that Gompertz's law makes a clear prediction about. Does the daily risk of dying in this species rise with age? The answer was no. The daily probability of death in adult naked mole rats, the analysis showed, did not rise exponentially. It did not rise gradually. It did not, by any statistical measure the team could apply, rise at all. From roughly 6 months of age onward, the age at which these animals reach reproductive maturity, the daily chance of any given naked mole rat dying in the next 24 hours stood at approximately 1 in 10,000. At 5 years of age, the figure was approximately the same. At 10 years, the same. At 15, the same. At 20, the same. The animals that survived past the age of 25, and a significant fraction of the population did survive past 25, continued to die at roughly the same daily rate they had been dying at when they were juveniles. This is not what mammalian aging looks like. The exponential climb of mortality risk with age, the rising slope that defines what aging is in the mathematical sense, was absent. The animals were not dying faster as they got older. They were dying at the same rate they had been dying at when they were 5 years old, and they continued doing so for as long as the researchers had data on them. In some cases, more than 30 years of continuous observation of the same individuals. The implication, when stated as cleanly as the data allowed, was that the naked mole rat is the first mammal known to biology in which the standard mathematical pattern of aging does not apply. The aging that other mammals undergo, the slow accumulating physiological deterioration that drives the rising mortality curve, was not happening in any way that the demographic measurement could detect. The species was, in the language used in the paper itself, demographically non-aging. The result was met with some skepticism at first, in the way that any genuinely unusual finding in biology should be. The most careful critics raised reasonable questions. The dataset, while large, was drawn entirely from captive animals. The oldest individuals in the analysis, while remarkable for their age, were still a small subset of the total population. There was, in principle, the possibility that aging was happening, but at such a delayed onset that even the oldest animals in the dataset had not yet reached the steep part of the curve. The mathematics might be hiding a real but late-arriving senescence. The team kept collecting data. In 2024, in the journal Geoscience, the same group, joined by Thomas Park, whose Chicago laboratory by then maintained the largest naked mole rat colony in the world, alongside Buffenstein, who had relocated her work to the University of Illinois at Chicago, published an updated analysis. The dataset had doubled. More animals, more years of observation, more deaths. The result held. Across an even larger sample, with the oldest individuals now approaching 40 years of age, the daily mortality risk continued to show no measurable rise with age. The non-aging finding had survived its first major replication. In a separate review published in the same year, in the Journals of Gerontology, Buffenstein and her colleague Vince Amoroso wrote in language unusually direct for a scientific paper that the naked mole rat appears to be the only species known to date that defies the Gompertzian laws of mortality. The careful hedge, to date, is the right hedge given the still narrow base of the data set. The underlying claim, however, is the claim that the data now supports. What is happening physiologically to produce this pattern is also worth describing carefully. The non-aging finding is a statistical claim about the population. The underlying biology, what is going on inside individual animals to keep the daily death rate flat across decades, is a separate and equally interesting story. The picture that has emerged from the laboratory work is one in which the standard signatures of mammalian aging, the ones that contribute to the rising mortality curve in every other species, are either absent or sharply attenuated in this animal. Cardiac function, measured carefully across animals of different ages, shows no significant decline between the ages of 2 and 24. The hearts of old naked mole rats pump as well as the hearts of young ones. Bone density, body composition, metabolic rate, vascular function. Across all of these measures, the laboratory data show essentially no change between the third decade of life and the first. Neurogenesis, the production of new neurons in the brain, continues to occur at low but measurable levels well into the third decade. The reproductive system shows no equivalent of menopause. Queens in their 30s continue to bear healthy litters of pups at intervals not measurably different from the intervals at which they bore litters in their first reproductive year. The animals do not appear to be experiencing the slow internal degradation that, in any other mammal, would by the same age have produced obvious cumulative effects. There is one phenomenon that does occur with age in this species, which is worth mentioning to keep the picture honest. The skin loses some of its elasticity. Photographs taken of the oldest captive animals show wrinkling and translucency that are not present in younger animals. Whether this represents a true aging signal or simply the consequence of long exposure to particular environmental conditions in captivity is, at present, not entirely clear. It is, in any case, a minor visible change in an animal whose internal physiology shows, by every available measurement, remarkable constancy across decades. The mechanisms that produce this constancy are the subject of intense ongoing investigation, and several of them have already been touched on. The cellular machinery is unusually well maintained. Protein quality control, the system that catches and corrects errors in protein synthesis, runs more cleanly across the lifespan than in other mammals. Mitochondrial function, the production of energy at the cellular level, is preserved with age. The DNA repair mechanisms, including the CGAS-mediated mechanism that the Tongji group characterized in 2025, work more efficiently than in mice or humans. The inflammatory milieu of the body, which in other mammals drifts toward a chronic low-grade inflammation that is now understood to drive many aspects of the aging process, remains in this species more like the inflammatory state of a young animal. The combined effect of these maintenance systems, distributed across the body, appears to hold the physiological state of the animal close to its young adult condition for the entire span of its life. What this amounts to, in the picture of what the naked mole rat actually is one further dimension of the strangeness that has been accumulating across this episode. The animal that does not properly regulate its body temperature, that does not feel certain chemical pains, that survives without oxygen by switching to plant-style metabolism, that almost never develops cancer because of an unusually long molecule it secretes between its cells. This same animal does not, by the most basic demographic measure available, undergo the aging that every other mammal undergoes. It lives a long life. It lives that long life at a daily mortality risk that does not rise across the decades, and when it eventually dies, the cause of death is often not, in the conventional mammalian way, the accumulated downstream failure of an aging body. What that cause of death is, in the careful records of the laboratories that have followed these animals across their decades-long captive lives, is one of the genuinely unresolved questions in the field. Most of the deaths, when carefully autopsied, are not attributed to any of the standard causes of mammalian senescence. They are attributed to specific local events, infections, injuries from intra-colony conflict, occasional metabolic failures, that in another mammal would be part of a wider pattern of age-related decline, but that in this species appear to occur as isolated incidents against a background of otherwise preserved health. The animals do not, in most cases, die of old age. They die of something specific, often something that could in principle have happened at any age, and they die in a body that, until the moment of that specific event, was operating at the physiological level of a much younger animal. One of those animals, more than any other, has come to stand for the long arc of this finding. He spent 30 years in a single laboratory, beginning his life in a colony collected from a savanna in Kenya in 1980, and ending it as the dominant breeding male of the colony that had been built around him in San Antonio, Texas. He was, by every measure that anyone bothered to take, biologically remarkable. And by the time he died in 2010, he had outlived several of the buildings he had been moved between. On the morning of the 5th of June in 1980, in a stretch of dry savanna near the village of Larata in northern Kenya, a team of researchers led by Jenny Jarvis spent several hours opening a section of underground tunnel and recovering the animals that lived inside it. The work was difficult, slow, and dusty. The temperature at the surface by mid-morning was already past 30 degrees Celsius. The soil was hard, and the tunnel system extended in several directions through it, branching and curving in the way that all naked mole rat tunnel systems do. The animals had to be coaxed or caught one at a time, lifted out of the soil with care, and placed in transport containers that would carry them, eventually across an ocean. One of the animals lifted out of the ground that morning was an adult male. By his body mass at the moment of capture, the researchers estimated his age at somewhere between 1.5 years and 2 years. He was, by every visible measure, a typical worker. Pink, wrinkled, with the small eyes and the large front teeth that defined the species, and with no markings or features that would have made him obviously distinguishable from any of the other animals being recovered from the same colony. He was placed in a container, transported with the rest of the colony to Cape Town, settled into Jenny Jarvis's laboratory at the university there, and given the kind of routine identification, a number, a record of capture, a date, that a research animal receives when it joins a captive colony. He was, at the time of his capture, the same age as a college sophomore. He would still be alive when several of those sophomores had retired. The animal was eventually moved out of Cape Town when Jarvis's collaborator Rochelle Buffenstein took a position at City College in New York in the late 1980s and brought a number of naked mole rats with her to establish a colony in the United States. The animal traveled north with that colony. He was by then somewhere in the middle of his first decade of life. He showed, on every available measurement, no decline of any kind. He was eating normally. He was active. He was integrated into the social structure of the colony, eventually rising, in the way that older male mole rats sometimes rise, into the position of one of the breeding males, the small subset of the colony that mates with the queen. In the late 1990s, when Buffenstein moved her laboratory again, this time to the Barshop Institute for Longevity and Aging Studies at the University of Texas Health Science Center in San Antonio, the animal moved with her. By then, he was 20 years old. By the standards of any other rodent, he had no business being alive. A house mouse lives, on a generous estimate, around 3 to 4 years. A brown rat lives somewhat longer, in the right conditions, perhaps to 5 or 6. A capybara, one of this animal's closer evolutionary relatives, lives a little over a decade in captivity, occasionally 15. The animal that had been lifted out of the Kenyan soil in 1980 had, by the year 2000, outlived every other rodent of comparable body size by a factor of 4 or 5, and he was, by every measurable standard, still operating as a healthy adult. He was by this point known to the laboratory staff as Old Man. This was not a formal scientific designation. It was the kind of name that small captive colonies of long-lived animals tend to accumulate over time, an affectionate working label that the people who cared for him used among themselves. The colonies in the Buffenstein laboratory were large, with many individual animals, and the practice of giving each animal a single distinctive name was not, in general, feasible. But there were a small number of animals who, by virtue of their age or their position in the colony or their personal history, acquired names anyway. The male who had come out of Kenya in 1980 was one of them. He remained the dominant breeding male of his colony into the first decade of the new century. He remained, by every available physiological measurement, an animal whose body was operating at the level of a much younger naked mole rat. The vascular function was intact, the bone density showed no measurable decline, the reproductive system was still functional, he was still mating with the queen, and the queen was still producing litters. His muscle mass had begun in the last few years to show some loss. He had become, in Buffenstein's own description, sarcopenic, the term used in human medicine for the progressive loss of muscle that accompanies aging. He had begun to look slightly like an old animal. But the change was gradual, and it had not begun until he was already in his late 20s, at an age, in mammalian terms, that was already entirely outside the range of what any other rodent of his size could reach. He died in 2010. The cause of death was, as is often the case in this species, not entirely clear. He had not developed cancer. He had not undergone the kind of cardiac decline that, in laboratory mice or in human patients, would have produced the signs of approaching mortality that animal care staff and clinicians know how to recognize. He had simply, over a period of a few weeks, slowed down, and then, one morning, he was gone. At the time of his death, he was the dominant male of his colony. He was, in Buffenstein's words again, the first to feed when food was brought into the enclosure. He was, possibly, still functioning as a breeding male, although by that point his breeding activity had become difficult to verify. He had spent 30 years in captivity. Added to the 1.5 or 2 years of life he had lived in Kenya before his capture, his total lifespan was, conservatively, just over 31 years. He was, at the moment of his death, the oldest naked mole rat ever documented under continuous observation by science. The record was not held in any official sense. There are no official records for the lifespans of laboratory rodents, of the kind that exist for human supercentenarians. But the dataset that Buffenstein and her team had been maintaining since 1980 made the claim straightforward to verify. No other naked mole rat had been continuously observed from a known starting age for as long as this one had. The body of the animal was, at the moment of death, in some senses the most valuable specimen in the colony. Autopsy and tissue analysis followed standard laboratory protocols. The samples were stored, catalogued, and made available to researchers across the wider naked mole rat scientific community. Whatever could be learned, biochemically, about a body that had operated for 3 decades at the physiological level of a much younger animal was learned from those samples and from the samples of his colony mates in the years that followed. Some of the molecular findings discussed in the previous chapters of this episode were derived in part from animals whose breeding history traced back to the Kenyan colony of 1980 and to the male who lived for 30 years as a stable feature of that colony. The animal had, across the course of his life, occupied 4 laboratories on 2 continents, been transported across the Atlantic Ocean, lived through 6 American presidencies, outlasted the Soviet Union, predated the public internet, and predated almost every piece of biomedical technology in routine use at the time of his death. He had not, in any meaningful sense, ever left his colony. The colony had been moved several times around him. He had remained throughout in the same general structure of warm bodies, dark corridors, and steady reproductive hierarchy that the colony had maintained since its founding. The world above his enclosure had changed considerably. The world inside his enclosure had not. The case of Old Man is, in some ways, a single data point. He was one animal. He died in 2010. The colony he was part of has continued without him, as captive colonies do, with newer animals taking his roles and the genetic lineage continuing through the queens whose litters he had once helped produce. His significance in the scientific literature is not as a record holder. There is now, in the Buffenstein Laboratory's care at the University of Illinois at Chicago, at least one animal older than he was at the moment of his death, a male whose age has been verified at approximately 39 years, who is still alive, still active in his colony, and who is on track to push the documented maximum lifespan of this species beyond 40 years for the first time. He will, when he eventually dies, be subject to the same protocols of tissue collection and autopsy that were applied to Old Man, and his body will be added to the same long accumulating dataset. The significance of Old Man, then, is not in being the first or the last. It is in being one of the early proofs in a small captive colony in a series of laboratories that the demographic finding described in the previous chapter was something other than a statistical artifact. The flat mortality curve, the preserved cardiac function, the continuous reproductive activity into the third decade, the absence of the standard signatures of mammalian aging, all of these claims rest ultimately on the bodies of individual animals, and Old Man was one of the bodies on which those claims were built. He was a small pink mammal who lived in good health for 30 years in a series of plastic containers in a series of laboratory buildings in the United States, beginning his life in the dry red soil of Kenya at a time when the field of comparative aging biology did not yet, in any meaningful sense, exist. He never knew the science. He was, throughout, what he had been when he was captured, a member of a colony, eating tubers, contributing to the work of the group, occupying his particular place in the social order. The aging of the rest of the mammalian world went on around him, in laboratory mice that lived 3 years and died, in laboratory rats that lived 5, in successive cohorts of researchers and students and animal care staff who came into the laboratories where he was kept, did their work for a decade or so, and moved on. He stayed. The colony stayed. What he and his colony had been quietly proving across those decades was not yet quite known to the people working with them. The molecular machinery, the CGAS variants, the hyaluronan signaling, the fructose metabolism, the dampened inflammation, the calcium response, the ribosome quality control, would not be characterized for another 20 or 30 years after his capture, the picture being painted in real time by his body and the bodies of his colony mates was a picture whose meaning would not be assembled until long after most of the early colonies were gone. The picture is still being assembled. In October of 2025, in the same journal that had published Park's 2017 anoxia paper, a research group at Tongji University in Shanghai reported a finding that none of the earlier work on this animal had predicted. The group, led by Ying Jiang and Jiong Mao, had been working for several years on a protein called CGAS, cyclic GMP-AMP synthase, the central detector in one of the cell's basic alarm systems. In every mammal that had previously been examined, CGAS sat in the cytoplasm and the nucleus of the cell with a dual function. When it encountered foreign or damaged DNA, it triggered an inflammatory response. When it sat near the sites of ongoing DNA repair, it suppressed one of the major repair pathways, an inhibition that was now understood to contribute to the accumulation of unrepaired genetic damage that drives mammalian aging. The Tongji group had sequenced the CGAS protein from the naked mole rat and compared it with the CGAS proteins from mice, from humans, and from a range of other mammals. They found, in the relevant region of the protein, 4 amino acid substitutions that distinguished the naked mole rat version from every other version on record. 4 substitutions out of a protein several hundred amino acids long. A change that, in proportional terms, was almost vanishingly small. When they tested the functional consequence of those 4 changes, the result was unexpected enough that they spent the next several years confirming it. The naked mole rat version of CGAS did not suppress DNA repair the way the mouse or human version did. It supported it. The same protein that, in every other mammal examined, acted as a brake on one of the central mechanisms of genome maintenance was, in this lineage, no longer a brake. The 4 amino acid changes had altered the way the protein interacted with the rest of the repair machinery, leaving the repair process freer to operate, and the cells of the naked mole rat with a measurably stronger capacity to fix the kinds of double-strand DNA breaks that, in ordinary mammals, accumulate across a lifetime. The team then went further. They inserted the naked mole rat version of CGAS into the cells of mice. The mouse cells, carrying the new variant, showed improved DNA repair. They inserted it into the cells of fruit flies. The fruit flies carrying the new variant lived longer. The molecular substitution, isolated from the body of the animal it had evolved in, was sufficient on its own to produce a measurable shift in the cellular condition of 2 species separated from the naked mole rat by hundreds of millions of years of evolutionary history. 4 amino acids. That is the size of the change. This is the most recent piece of mechanism to be added to the picture of why this animal lives the way it lives, and it is, by some distance, the most evolutionarily peculiar piece so far. The protein in question is highly conserved across the animal kingdom. The CGAS of a mouse and the CGAS of a human are very nearly identical, and both of them are very similar to the CGAS of essentially every other mammal that has been examined. The naked mole rat lineage has, somewhere across its 31 million year separation from the rest of the mole rat family, accumulated 4 specific substitutions in this protein that converted it from a hindrance to genome maintenance into a support for it. The substitutions are small. The functional consequence when measured in the cells of the animal is large, and the implication, in the context of everything else this episode has tried to describe, is that the strangeness of this animal is being produced by a long series of such small specific changes, distributed across many genes and many tissues, each of them individually subtle, but cumulatively sufficient to produce the body that lives under the dry soil of East Africa. The picture that has emerged across the past 2 decades of work on this species is a picture in which the unusual traits, the cold-blooded thermoregulation, the indifference to chemical pain, the plant-style fructose metabolism under anoxia, the long-chain hyaluronan in the extracellular matrix, the 4 amino acid changes in CGAS, are not produced by any single master mechanism. There is no one gene, no one protein, no one regulatory shift that explains the whole animal. There is, instead, a long accumulating list of specific, individually characterized changes, each of them identified at the molecular level, each of them now in the early stages of being studied, and at least one of them already in the process of being transferred into the cells of other species. The animal that has been producing these findings does not, in any meaningful sense, know that any of this is happening. The colonies in Chicago and Berlin and Rochester and Tokyo and Shanghai continue their daily routines. They dig their tunnels. They eat their tubers. They suppress one another's reproductive cycles. They raise their pups. They sleep in piles of warm bodies in chambers that hold their internal climate steady year after year. Whatever the laboratories of the world have learned from them, and whatever the laboratories of the world will go on to learn, the animals themselves have not changed at all in the time the learning has taken. There is a question worth asking in the quiet that follows a list of findings like this one. The question is what it means, in the broader picture of biology, that one mammal has managed this. The naked mole rat is a small, restricted, evolutionarily peculiar species. It lives in a particular kind of habitat, occupies a particular kind of niche, and represents a single branch of a small family on the wider mammalian tree. By every standard measure, it is an exception. By the count of total mammalian species on the planet, somewhere around 6,500, the naked mole rat is one of one. The traits it carries are not, on any current evidence, going to spread through the rest of the mammalian lineage. they are confined by the long evolutionary history that produced them to this single small line of subterranean rodents. But the traits exist. They are not theoretical. They are not predicted by models. They are not extrapolated from the behavior of laboratory mice. They are present in the body of a living mammal, available for laboratory study, and now, in at least one case, transferable across species lines. The argument that has often been used in the field of medicine, that the standard human diseases of aging are simply the unavoidable consequence of being a mammal with mammalian cells in a mammalian body, has, since the careful characterization of this species, become slightly harder to defend. The diseases are not, evidently, unavoidable. They are common, they are widespread, they are present in every other mammal that has been studied in sufficient detail. But in this one species, they are nearly absent. Whatever the standard model of mammalian aging actually is, it is not a fixed property of the underlying biology. It is a state that, at least once in evolutionary time, has been substantially modified. What that modification might mean for the broader project of human biomedicine is a question that will not be answered in this episode. The translational work is in its early stages. The mouse experiments that have been completed are encouraging without being decisive. The path from a mouse experiment to a human treatment is, as the previous chapter discussed, long and frequently disappointing. What can be said with some confidence is that the existence of this animal, the simple fact that a small pink rodent of East Africa has, across the long action of its evolutionary history, arrived at a set of biological solutions that the rest of the mammalian world has not, represents a kind of proof of concept. The solutions exist. They are reachable in principle by mammalian biology. They have been reached at least once. Whether they can be reached again by any route other than the slow accumulation of millions of years of natural selection is the question the laboratories of the world will spend the coming decades trying to answer. The conservation status of the species itself, set against the scientific attention it has attracted, sits in an unusual place. By the standard measures of population biology, the naked mole rat is not at risk. The International Union for Conservation of Nature classifies it as a species of least concern. The animals occupy a wide range across the Horn of Africa and parts of Kenya. The populations are stable. The habitats are largely unsuited to large-scale human agriculture, and there is no immediate prospect of population decline. The species is not, in the language of conservation, threatened. What threatens it, to the extent anything does, is climate. The dry conditions that produce this animal and that continue to support it are sustained by a particular pattern of seasonal rainfall in the Horn of Africa, and that pattern is, on the available evidence, shifting. The tubers on which the colonies feed depend on the rains. The colonies depend on the tubers. A slow drying of the region, of the kind that climate models project across the coming century, would reduce the food base, fragment the populations, and gradually erode the conditions under which this species has, for 31 million years, made its quiet living. The change, if it comes, will not come quickly. But the long stability that produced this animal is not, in the present moment, guaranteed to continue. There is one other recent finding worth mentioning before the chapter closes. In late 2025, a separate research group working from genomic and morphological evidence reported that what has, until now, been treated as a single species, Heterocephalus glaber, may in fact represent 3 deeply diverged lineages. One of those lineages corresponds to the standard laboratory animal that almost every research group in the world has been working with. The other 2, distributed across slightly different parts of the original range, are sufficiently distinct in their genomes and their skull morphology that the older taxonomic name Heterocephalus philipsi formerly described in 1885 but long treated as a synonym, has been resurrected and applied to one of them. Whether the unusual biological traits that have been characterized in the laboratory animal will turn out to be present in the other two lineages in identical or modified form is a question that has not yet been answered. The research community is, at the moment of this recording, beginning the work of finding out. The animal, in other words, may not be exactly what the laboratories have for the past several decades assumed it to be. It may be a slightly different animal, or three different animals, with overlapping but not identical biology. The work of the coming years will sort that out. Whatever it sorts, the underlying picture will not change in its broad outline. There is, in the soil of East Africa, a small pink mammal whose body has drifted across 31 million years of subterranean life into a configuration that the standard mammalian categories do not entirely accommodate. The configuration is real. The configuration is being studied, and the studying is producing a slowly accumulating list of specific molecular shifts, each of which says something useful about what a mammalian body can be made to do, given enough time and enough evolutionary pressure. There is a sentence that has been said in various forms by several of the researchers who have spent their careers with this animal. When pressed on what the long arc of the work has meant, what the accumulated findings actually amount to, the response tends to settle, eventually, on some version of the same observation. The naked mole rat does not, in any of the senses that the words usually carry, behave the way a mammal is supposed to behave. It violates the rules. It violates them quietly, in a small pink body that fits in a closed human hand, in a colony that lives 3 meters under the surface of a country most people will never visit, with no awareness of any kind that the rules it is violating are rules at all. There is something worth holding still for in that. For the entire span of mammalian evolutionary history, for 200 million years, since the lineage that would become mammals first separated from the lineage that would become birds and reptiles, the body of every mammal has run on the same basic principles: warm blood, constant body temperature, continuous oxygen consumption, a finite lifespan governed by the slow accumulation of cellular damage and the rising exponential curve of mortality risk. These principles have been for so long simply what mammals were. The species that built them, the lineages that diversified from them, the radiations that filled the continents after the dinosaurs were gone, all of them have operated within those same constraints. And then, in one small corner of the lineage, in a branch that turned away from the rest of the rodents 31 million years before the present, under particular ecological pressure in a particular kind of soil, the constraints quietly bent. Not all of them, not entirely, but enough of them in enough specific ways that the body that emerged from the bending is one that the standard description of a mammal no longer cleanly fits. The body is still a mammalian body in every phylogenetic and developmental sense, but the body has, in its physiology, in its biochemistry, in its demographic behavior across a lifespan that runs for 40 years, become something that the category was not originally built to contain. This is what the long study of this animal has produced, not a miracle, not a cure, not a slogan about immortality or invulnerability or any of the other words that the popular reporting has, from time to time, attached to it. Something more modest and in some ways more interesting. A demonstration in a small living body that can be held in a human hand that the basic constraints of mammalian biology are less fixed than they look. That given enough evolutionary time and enough specific pressure, those constraints can be partially renegotiated. That a single lineage, working alone in a particular underground niche for tens of millions of years, can produce a body that does several things its ancestors did not do, and that its more visible cousins above ground cannot do. The animal does not know any of this. The animal has never known any of this. Across 31 million years of evolutionary history and across the 4.5 decades of close laboratory study by curious humans, the naked mole rat has been, throughout, simply the animal it is: pink, small, lightly whiskered, cold to the touch on the outside, warm at the center of the colony, indifferent to the burn of acid and the burn of chili, capable of breathing air that would kill a person standing on the surface, capable briefly of breathing air that contains no oxygen at all, resistant to cancer, unwilling by every available measure to age, and entirely, completely unaware that any of this is in any way unusual. Somewhere tonight, under the dry soil of northern Kenya, in a tunnel 3 meters deep where the air is warmer and heavier than the air above it, a colony of this animal is asleep in a single pile of small, warm bodies. The queen is asleep at the center of the pile. The breeding males are asleep beside her. The workers and the soldiers are arranged around them in the loose, tangled geometry that has been the colony's sleeping posture for as long as the colony has existed. The carbon dioxide in the chamber is high, the oxygen is low, the temperature is steady. None of the animals are dreaming about any of this. None of them know that several thousand miles away, in a laboratory on the west side of Chicago, or in a laboratory in Berlin, or in a laboratory in Rochester, or in a laboratory in Shanghai, the cells of their relatives are being studied carefully, one molecule at a time, by people trying to understand how a body can be built to do what their bodies do. The colony breathes its slow, heavy air in and out together. Somewhere in that pile, perhaps near the center, is one animal who has been alive since before the youngest researcher in any of those laboratories was born, and who, on every available measurement, is functioning as a healthy adult. The lights in the laboratory chambers are dimmed for the night. The work resumes in the morning. Rest well.